researchers, Dr. Vijay Upadhyay sir,
बघते आता पुढचं तर चालू झालंय.
this topic. So I request Upadhyay sir, please share your.
मी तुम्हाला मेसेज टाकेन. शेवटे सरांचं आहे माझ्या आधी.
Thank you, sir. Thank you.
So welcome to all. First of all, I congratulate and wish my best for all the audience and all the stakeholders and all the team members who are directly and indirectly involved for this scientific event. As you understand that the tuberculosis topic cannot be covered in 30 minutes, the time which is allotted to me. Can I share my screen, sir? I shared my screen.
Yes, sir, you can share your presentation.
Is it visible? Hello? I request few of the speakers or somebody's background voice is there, which is doing the interference here. So as you understand that the tuberculosis topic cannot be covered in 30 minutes. Just now, the 24th March 1882, after the discovery of the Robert Koch, still after 100 years, we are still struggling for the novel, whether it is a vaccine or the novel drugs. After 100 years also, when I started my PhD, the WHO declared that in 2020, the TB can be controlled. But still, now WHO declared that TB is everywhere. What are the last 10 to 15 years of experience in my tuberculosis from my lab? I just mentioned few of the biomarkers here, as TB topic cannot be covered in 30 minutes. So I shall go fast. I shall not mention that still many techniques, whether the cultural, molecular diagnostics, direct methods, indirect methods, you can find out on the screen. Many, there are the many types of the tests are available, but still the culture test is still a gold standard. And obviously the gene expert and from Malbio is also one true PCR and the TrueNat test is there. Still they are the gold standard test, and other tests are you can call as a parallel test, which is going on in the tuberculosis. Now, when we are seeing that any patient when going for the doctor, there are the n number of the tests are there. Some are the high complexity assays, some of the moderate complexity assays, some are the lower complexity assays. Means I can say that apart from the culture, even if it is a gold standard, it is taking more time. So I just summarized out that there are the n number of the tests having some potential limitations, advantages, disadvantages. And still the TB is after the HIV and all the malaria, and still it is the major global scores. You can call it as still it is a challenging, giving the challenge to the scientists. So we cannot come out from this complexity. We can just call it out. It is a high complexity, moderate complexity, and the low complexity. Microscopy, fluorescent test, culture, gene expert, LAM, liporhabdomanian test, because we understand that tuberculosis high lipoic acid content is there. Because of that high cellular this lipid content, it resists most of the drug actions, and because of that the drug resistance, whether in the form of multi-drug resistant or the extensively drug resistant, is also coming into the picture. But still the TB, there is a n number of scientists working throughout the India, throughout the globe, but still we are still struggling for after 5 drugs, there is no other drugs came out. I'm appreciating on the note that the TrueNat and other tests are coming at the molecular platform, but these molecular tests are only can be applicable in the tertiary hospitals where the sophisticated things are available. Still the India, which is belongs to the rural population, there we need the grassroots test. So I'm taking you back to what is the understanding in the tuberculosis, why these challenges, because we understand when we are calling it as a drug resistance. So you can see that in India, there is a diversified, qualified doctor, unqualified doctor, qualified lab, unqualified lab, pharmacy, unqualified pharmacy. So whatever the drug resistance is coming, whether multi-drug resistance or the extensively drug resistance, it is because of the wrong practices of the other doctors. Because any more than cough, if cough is there, they are writing the rifampicin, they are writing something. So because of that wrong practice of the few doctors, I'm not bothering any discipline of the science or anything. But because of that, the resistance came. So because of this 20, 30 years of the wrong practice or more than so, now MDR, XDR is still the challenges for us, global challenges. So already TB is a problem. there is a multi-drug resistant, then XDR, extensively drug resistant. And also we understand that TB is not oriented to the pulmonary, to the lungs. There are extrapulmonary TBs also there, bone and joint TB, lymph node TB, abdominal TB, ocular TB, meningeal TB, miliary TB, cerebrospinal fluid, ascitic, even the gastrointestinal TB is there, genitourinary TB is there. So we can call it as the TB is everywhere in the body, also everywhere in the body. So how to come out with that? So should we again rely on the culture test? No. I'm taking you 10 years back when we used to take out some antigens from the culture filtrate. Why we are taking the culture filtrate antigens? Here I want to specify that, because one thing is very clear that after 100 years, 1882, still culture test is gold standard. So obviously we should dig, we should take out the water only from that source, which is a gold standard and it is widely accepted. So what we used to do, we are maintaining that MTB S37 RA culture, a virulent strain on the LJ slant. Then we are expanding this culture to 4 weeks. Everybody knows it is taking 1 month time for the growth. We are expanding to the Sawtons medium. This Sawtons medium we enriched with some thyroxine and some other hormone, and we reduced their growth to the 3 days to the 7 days. So in 3 days, in 1 liter, and then it is expanded to 5 liter, 10 liter, 7 days. So almost in 4 weeks time, whatever the growth we got it, we transferred into the Sawtons medium, liquid medium, and in 3 and 7, 2 subsequent phases, 10 days, we are getting culture medium. We are getting culture filtrate antigens. Now it is a question to purify these antigens. So we simply did that purification of antigen. But for that also we need to take the millipore assembly and all this, because ultracentrifugation, dialysis, these are the costly things, and taking time also, and infection is also main problem if we are potentially handling the MTB and RV. So what we simply did, earlier what we used to do, whatever the M. tuberculosis organism is there, we used to cultivate on that LJ slant. We just do the phenol inactivation, we do sonication, SDS, and instead of culture filtration, whatever the 5-10 liters culture, we simply taking the cells and we are making its sonication, and by means the SDS extraction buffer, we called it as a SDS, PISS buffer. What is PISS buffer? Phenol inactivated SDS soluble sonicate antigen. Now this is the source from where you can take out the many antigens. So what is the understanding behind this? Again, I'm referring that TB is a gold standard, culture test is a gold standard. Taking directly antigens from the culture filtrate, rather we discovered some technique that we took up MTB cells, phenol inactivated, sonicated, SDS extraction, and simply PIS antigen can be used as a source. Now, the brilliant paper was published in Diagnostic Microbiology Infectious Disease. I was one of the co-author into that. What we did, we have sorted out separate antigen as per the molecular weight, like 31 kDa. So we label it ES protein. ES means excretory secretory antigen. What is the beauty of ES secretory antigens? When we are inoculation of the M. tuberculosis organisms on the LJ slant is taking place. So these M. tuberculosis, they are secreting some antigens, they are secreting some protein. We isolated that antigens, we purified it, and we called it purified antigens as a excretory secretory, because antigens, they are excreted and secreted by this MTB organism. And when we run it on SDS page based on their molecular weight, 31 kDa, 41 kDa, we got different panels of the proteins, and we published. As I told that TB is not only with pulmonary, there is a pulmonary TB, extrapulmonary TB is also there. So what seroreactivity we studied, the ES31 antigen and their separate antigen, we studied their activity in various forms of tuberculosis, pulmonary TB, extrapulmonary TB, HIV TB infection. Even that paper was published in Asian Pacific Conference of Clinical Biochemistry in China, Beijing. So now what we observed that from our observation that ES31 is good in pulmonary form of tuberculosis and in lymph node tuberculosis. 41 kD antigen, we call it as a ES41, is good in bone and joint tuberculosis. ES36, that is ES38, 38 kD molecule is good in abdominal tuberculosis. Again, EST6 molecule is there, that is having the good cross-reactive, having the good potential in the lymph node form of the tuberculosis and some of the meningeal tuberculosis. So what we did, now we thought that why should we go for individual antigens? So first time we did the attempt that we made it's a cocktail. Cocktail means the equal proportion of all the antigens, whether ES31, ES41, ES36, EST6, equal combination can be done, and their seroreactivity in sputum pulmonary tuberculosis and with can be compared. And more than 79 to, you can see that key 79 to 91% that sensitivity we observe. And one more important thing is that whole world is focusing on antibody response. Any organism going in our body, it is developing some antibody, and we rely on the antibody response. But antibody response is depend on our immune reactions. It is, if suppose immunosuppression is there, HIV case is there. So in immunosuppression case, the antibody won't be get developed. To counteract that, we first focused on the antigen detection. Suppose COVID is going on our body, rather than focusing on COVID antibodies, please focus on the COVID antigen itself. So that's why whatever the antigens entering in our body, whatever the antigens, my voice is coming, sir. Hello? Hello? So whatever the antigens are going in our body, I should detect. If I'm audible?
Yes, sir.
So first time in India, what we did, the first time the free antigen form and the IC antigen, immunocomplex antigen. You always know that antigen antibody, they cross reacts with each other. So we are not only detecting the antibody, but we are detecting antigen also and the immunocomplex antigen also. In filariasis, in leprosy, the immunocomplex antigen things, immunocomplex antigen is IC. Hello? Some background voices coming. So see ES31 antigen as some background voices also coming. Anyway, so what my understanding is that when ES31 antigen, we have seen in the immunofluorescent form. In that immunofluorescent form, we also got the Mycobacterium tuberculosis is helping the Mycobacterium tuberculosis multiplication in axenic culture and in human macrophages. You always understand that whether it is the cancer study or in the HIV or anything, the cell line studies are very, very important. So what we need to do this, what we have done, the ES31 importance in cell line culture, and you can find that when ES31 antigen is released into the microbial cell line. So this paper was also published in the Scandinavian Journal of the Infectious Diseases. Sir, I request some other speaker voices also coming in the background. Hello? 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 Some background voices coming.
You are audible, sir.
Some background voices coming, sir. Some
noise. Yes, sir, you're audible.
So detection of antigen and the antigen in pulmonary tuberculosis. Now, as I told you that key antibody response is very predominant. So first time we focused on the free antigen detection and the immunocomplex antigen detection and all forms of extrapulmonary tuberculosis. What I did that key extrapulmonary tuberculosis, TB lymph node, TB meningitis, bone and joint tuberculosis, abdominal tuberculosis, pleural tuberculosis, genitourinary tuberculosis, miliary tuberculosis, ocular tuberculosis, all forms of the extrapulmonary tuberculosis were explored. And all these cocktail antigen, ES31, 41, ES43, all we made its equimolecular, the combinatorial form, and their free antigen detection as well as immune complex antigen detection we detected in extrapulmonary tuberculosis, and that paper was published in Biomedical Research Journal. So what is my intention to do that? If whatever the 5, 6, 10 years before we studied, it is real time, because in 2008, the serology was banned. When serology was banned, most of the good antigens having the good diagnostic potential were get sidelined. Now everybody is focused on the molecular platform, but really saying that in our India, there are very tertiary hospitals are very rare and very less. And if molecular platform, if laboratory, we need expertise, we need the PCR and all these things. Now you can see that we also published the Western blotting of this M tuberculosis infected patients, and from 16 kDa, 20 kDa, 43, 55, 66, 85, all these types of the antigens, we have found in the circulating tuberculosis antigen, sputum positive pulmonary TB sera, sputum pulmonary negative sera. So it is real time to explore all this. I'm having all these antigens with us. It is real time that somebody can come and help in developing these antigens on the immunofluorescence platform or the chemiluminescence platform. Now the fractionation of the CTA, the circulating tuberculosis antigens, from 14, 12, 14 kDa to the 160, 95. You understand that the molecular weight range of the protein, we have covered almost all the proteins. So not only the 31, 41, 43, EST6, CTA antigens like the circulating tuberculosis, because we observed that some antigens, they are circulating in the blood of the patient. So these type of the novel studies are nicely published in, yes, International Journal of TB and the Lung Disease. I am one of the co-author. I told you that some of the good marker, ES31 and the ES20, like the 20 kDa molecule ES20 and 31 kDa ES20, that we observed in the lymph node tuberculosis, and you can find that immunocomplex antigen, most of the people, they are focusing only on the antibody and the antigen. We first time focused on the immune complex antigen detection, and it was published in International Journal of TB and Lung Disease. The ES20 antigens, we also find it good in the lymph node tuberculosis and some form of the pulmonary tuberculosis. You can find the Western blot studies here. So what these Western blot studies is there? It is time to pull all these antigens into one another, and now we can make it into the next form. So tuberculosis, some ceremony is going on. Hello? Some inaugural function is going on, or shall I continue?
Yes, sir. You continue, sir.
So now I want to focus on the London study. What is my London study? In that London study, we focused more on the TB exposure. I just request that somebody can mute that background sound of the inauguration is coming. So I request who is in charge of this session, I think mute, because it is creating the disturbance in the presentation. So in tuberculosis, main is very important thing is that we belongs to the endemic country. So what we are doing in the endemic country, when we are in the endemic country, we are prone to the TB bug. So TB is very common. So like COVID, now we are all exposed to the COVID atmosphere. So many people are telling you are infected or uninfected. I'm happy to tell that infected, uninfected, but I want to add one more word, that we are exposed to the TB environment. So based on exposed to the this environment, MTB environment, in our body, there is an interferon gamma response will be there. That interferon gamma response will come positive indirectly. So what need to do that? This is the Hinduja Hospital study in collaboration with the Imperial College London, that national TB. First time we focused on the whole blood gene expression studies. So what this is telling that M tuberculosis, they are having particular region of difference gene, that RD1 gene, which is not shared by other environmental bacteria or the BCG strain, the Bacillus calmette-guerin. So it is time to focus on that RD1 gene, ESAT6 and the CFP10. They will only focus upon the exposure level. They will only focus on the infection level. So this is the T-spot study that I developed in the Imperial College, and that ESAT6 we got good results. This is the ELISpot test, simple ELISA test is there, where the Nunc microtiter plate is there. Only in the simple ELISA, what we do, we used to do 2.5 hour ELISA we used to do. Here the ELISpots are there. So one spot corresponds to the one antigen response. So in that way, there are the counter, there the cell counters or the ELISpot counters are there. So after the ELISpot result, whatever the results you will get it, that results will be counted by this automatic counter. We call it as a T-spot TB test or the ELISpot test. So when I was working at the Immunoshop India, we developed first time of this kit, that the T-spot TB, and the quantiferon TB is also known. Most of the people telling that it is not differentiating latent to active TB. But as I am telling that today we are focusing only on the tuberculosis, and culture is a gold standard. So after that culture and the gold standard, what is the other parallel test we are having to test the latent TB? The latent TB is just like a Titanic iceberg. Whole world is focusing on that TB, but nobody is focusing on that. The latent means the dormancy. Same like in HIV TB, there is a dormancy is there. Same like in other things, there is a dormancy is there. Window period, you can call it as that. So IGRA, interferon gamma release, it is a real time to develop the cytokine. So what is my focus? Whatever the ES31, 43, EST6, 41 KDA I am telling, along with that ESAT6 and the CFP10, all to be come under the CLIA platform, where in 20 minutes, the TB can be detected in all forms of tuberculosis, not only with the pulmonary TB, but extrapulmonary TB and HIV TB co-infection. It is just overnight study that you can take the sample of the blood of the patient and you sensitized with these antigens, and next days after 24 hours, you can measure its interferon gamma status by the standard curve or the platelet. So we conducted this type of study in collaboration with the BJ Medical College, National AIDS Research Institute, NARI, and this project was with the Johns Hopkins University, USA also. And we got impressive results of the 95 and more than this. The paper was published also here. So moral of the result is what? It is not a time to focus on only one thing, this is good. Whatever the good thing is there, we have to come and we have to come out with the good diagnostic tool molecule. After 2008, when the serology was banned, everybody is telling that what are the novel biomarkers there, they were sidelined. But my request is that, my appeal that, though the serology is banned, you can boycott the ELISA, but let other platforms are there, immunofluorescence, chemoluminescence, means molecular also you can go side by side, but let other things also to go on. Other things going on means we have to go on the PCR. Along with the PCR, there is a chemoluminescence and there is a immunofluorescence test should also be going on. Now you understand that what are the antigens I quoted, ES31, ES41, ES38, ES43, CPET, all they have importance not only in pulmonary tuberculosis but in extrapulmonary tuberculosis, whether TB, uveitis, laryngeal TB, POTS disease, inflammatory rheumatic disease, juvenile idiopathic arthritis, inflammatory bowel disease, hemodialysis patient, TB meningitis, TB peritonitis, erythema, enduratum, TB cirrhosis, TB pleurisy, and also in comparison, most of the time intestinal TB is same from the Crohn's disease. So these tests help in differentiation. The TB can be differentiated from sarcoidosis lymphoma by this. So these are some of the outcomes of the IGRA results, how they are giving the sensitivity more than 90% and specificity 95 to 99%. So what is the future directions are needed? The future directions are needed is the RD1 genes, which is only conserved in MTB virulent strain. The M tuberculosis or MTB virulent RD1 region of difference is present, where the new age antigens RB2654, 3615, 3879, 3873, 3878, all these culture filtered antigens, inducible proteins, monocyte chemotactic proteins, they are need to be explored on the chemoluminescence platform, also on the immunofluorescence platform. There are the interferon gamma response, there's memory cells, interleukin 2, human necrosis factors also there, cytokine signatures also there, biomarker discoveries also there, proteomics and transcriptomics there. So now it is time that we are focusing, as we got all the antigens which are having the diagnostic potential in ELISA, Western blot, and we explore on the immunofluorescent also, that I showed you the immunofluorescent style. Now it is a time to focus it on the chemoluminescent platform, the CLIA platform, where you can, everything is same like the ELISA, the antigen antibody reaction, only the probe will be different. Even HRP will also be there, but you have to add the luminol substrate and you have to just incubate 10 minutes, and within at 37 degrees Celsius in 10 minutes, based on the relative light unit, you will get the fluorescence result, the chemoluminescent results also. So chemoluminescent testing, which is help in diagnosing antibodies also. The magnetic beads are simply there, they can coated with these ES31, 43, 46 and EST6, and the sample can be added. And when the tracer will be used, the antibody labeled with isoluminar, it will trigger the relative light unit, and with the light emission, there will be the results will be observed. Now the fluorophore or the antibodies also there. As I told you, whatever the antigens responsible or useful in the ELISA, they can be explored to chemoluminescence also or the fluorophore also. So immunophore can also be. So this is my talk. I don't know who is listening or not. Hello. As 30 minutes given to me, so I wind it up. Hello.
You are audible.
There is alternate ceremonies going on, some inaugural function.
This is the only function is going on.
Hello.
Sir, you are audible, sir.
Any question? I'm asking any questions.
Thank you very much, sir. Thank you very much, sir. Now the topic is open for discussion. क्वेश्चन तिकडनं विचारलं तर चालेल का? हे ऑन आहे सगळं? कुठलं विचारलं तरी चालेल सर. Is audible? Kadam sir is audible?
Yes.
Hello.
Yes.
Hello, Upadesh sir.
Yes, please.
Yes. Actually, initially you mentioned within 30 minutes this topic can't be finished. Still, you are given a broad outline and very informative presentations to the all audience who are attending your presentation online as well as offline mode. Actually, we are running PG programs in microbiology and biotechnology. And actually, we can't more expose to our students in medical microbiology or medical biotechnology. Actually, we were expecting you should be in Nanded, but due to your busy schedule,
No, I will come. I will come.
still you are given time for online presentations. So on the behalf of the audience, I am very thankful to you. From audience, I have one question, sir. Hello, sir.
Yes.
Actually, best detection methods you have mentioned now for tuberculosis, but many times what happens in rural area or even if in urban area also, right times we can't detect the tuberculosis, and in such patients they are immunocompromised, immunosuppressive, and due to that, multiple drug resistant microorganisms establish infections, and many patients remain in danger. Okay,
Yes.
so such techniques, if available in the government hospitals, it will be more important or more advantageous to the people also. Okay, actually your work is very, very interesting, and as I have mentioned, you should give the more time to us in form of the other lectures also. So thank you
30
very much.
minutes. TB cannot be covered in 30 minutes. And as I'm telling you that 2 patents we filed, where the 1 month growth we have come down to 10 days. And 14 days on liquid culture, we reduced it to 48 to 72 hours. That powder or that component we have developed in that culture, and we patented it also. We are looking for its commercialization. So it will be helpful for the rural people and also for the we don't have access to the tertiary hospital.
Okay. So on the behalf of the organizing committee, I'm very thankful to Upadesh sir. Thank you, sir. Thank you very much, sir. Thank you. Next.
Thank you very much, sir. Now, ladies and gentlemen, we are rarely get this kind of opportunity to listen the words a knowledgeable speaker, Dr. Yogesh. I would like to request Dr. A.P. Pathak ma'am to please introduce or Dr. Yogesh.
Good afternoon.
Please stop your screen sharing.
Good afternoon, one and all. May I request Honorable Shauchi sir, please turn on your video and.
It's on, madam. My video is on.
Thank you, sir. So, the task of introducing a great eminent speaker and guest of honor is great privilege for me. Dr. Yogesh Shauche sir, PI, Ncmr, NCCS Pune, and Professor Azim Premji University, Karnataka. Before I formally introduce sir, I wish to express my sincere thanks for help and guidance that sir have extended to School of Life Sciences in various capacities. I'm really happy to introduce an eminent, very simple and great scientist having diverse and huge credentials in his name. I agree that sir don't require any introduction. However, for this new generation, at least the students should know the credentials that you have. So please allow me, sir, to introduce you hardly for 2 to 3 minutes. Dr. Yogesh Shauche sir started his career as scientist, National Center for Science, NCCS in Pune, India. After working on different aspects of microbial, especially bacterial diversity of various ecological niche, his thrust area of research is now to understand succession of microbial communities in human gut during development. He is working for more than 25 years in field of microbial ecology, microbial molecular taxonomy and biodiversity. Published more than 200 publications in journals of relevant area, reviewed publications in international journals like FAMES Ecology, Scientific Reports, International Journal of Systematic and Evolutionary Microbiology, and Microbial Ecology. Sir have also publications in this journal, and for your information, these journals can be named as dream journals for researchers. He is also on editorial board of prestigious journal like Current Science, European Journal of Soil Biology, PLOS ONE, and Scientific Reports. 2-3 pages of information I have with me, but I will not read all those 2 and 3 pages. Just I'll try to explain you that very eminent speaker and scientist we have. And now I request Shauche sir. Sir, you please deliver your talk.
Thank you very much, madam, for this opportunity. And I would like to share the screen. Okay. Yeah. So, can you see the presentation now? Yeah, you can.
Yes, sir.
Yeah, and yeah, now it has gone to full screen also. Good, great. Yeah. So indeed, it's a great pleasure to be here and talking to you all. And going by the, I love to come to Amravati, and I would have liked to deliver this talk in person, but because of the travel constraint and all, I could not come. Otherwise, I would have loved to give this talk in person. Going by the theme of this particular symposium, what I'm going to talk to you in next half an hour, 50 minutes is about how the microbiome, or in general, microbes could be a source of bioactive molecules, and what are the recent trends, what are the new things that have come up in discovering novel bioactive molecules from microbes. So I'll give you some background also, so you'll have to excuse me if I'm going too basic in my initial part of my talk, but I thought since the crowd is largely students, it is important that I talk about this background. So microbes have been a very popular source of bioactive compounds right from the beginning. We all know that majority of antibiotics have come from microbes, and after penicillin, several antibiotics have been discovered from microbial sources. So that we all know. As the science evolved, the technologies for extracting exploring microbes for bioactive compounds also changed. So essentially, the way people started using multiple approaches. So essentially, whatever sample you have, one way is the standard way you do isolations, and then isolates you use for bioactivity screening, and then you do standard structural assays, dereplication activity, and then if you find some, whatever activity you are looking for, then it goes through further drug discovery pipeline. I will not talk about this pipeline and all. That is a time for some other talk, or you may be already aware of that. But with the advent of DNA-based technologies and the whole issue of 99% of bacteria being not cultured, people started using DNA-based approaches also, because to cut the long story short, most microbiology students would know that only 0.1% microbes from any environment can be grown in the laboratory, and 99.99% remain uncultured. And these bacteria that could not be cultured in the laboratory are a vast source of bioactive molecules. So in order to screen for them, then this DNA-based approach was developed, where you generate metagenomic information. Primarily, this is a sequence information. So you have the DNA, you do a massive sequencing of this DNA, generate the sequence data, and then you generate information about diversity, physiology. You can use that information also to grow some of the organisms in culture. So when you have the sequence information for an organism, you can know about its physiology, and knowing the physiology and nutritional requirements, you can design the media to grow that organism. And then again, these organisms, you can grow them and do the screening. And some of you may remember the story a couple of years back when a novel antibiotic called Teixibactin was discovered using this approach. And that is the antibiotic which is active against all drug-resistant organisms, and it is unlikely to develop any resistance for next 30 years. So that is the power of metagenomics. There is another way also that is based on purely sequence information. Even if you do not have the isolate in culture, based on purely sequence information, you can look for novel bioactive molecule producing genes. These are called as biosynthetic gene clusters. So this will depend on your knowledge you have for the biosynthetic pathway for a particular compound, and it is very easy for the antibiotics. So lot of novel antibiotics, people screen the metagenomic data for known biosynthetic gene clusters and arrived at many novel molecules. And then again, it goes to the same pipeline. The isolates also, you can do the genomics and do the same, like get the genome sequence of the isolates. Instead of screening, first get the genome sequence, then look for any novel compounds in the genome sequence. So that is the protocol which nowadays followed for looking at novel bioactive molecules from bacteria. Coming back to situation in India, some of you would know that my earlier organization, National Center for Microbial Research, is itself born out of such massive exercise taken in India, wherein isolates were collected from diverse ecological niches, and they were screened for certain bioactive molecules. So these bioactive molecules are in the range of anti-diabetic, anti-infective, anti-inflammation, and anti-cancer. So under these four categories, the biomolecules were screened, and this led to the repository of over 200,000 microbes across the collected from diverse ecological niches across the India. So the overall pipeline of this project was something like this, that a large number of over 250,000 isolates were collected. The screening was done for anti-cancer, anti-diabetes, anti-inflammation, anti-infective. which was done by Piramal Life Sciences that time. The leads were obtained in the first phase, and in the second phase of the project, some of these leads were taken further for chemical characterization, dereplication, and determination of molecular structure. So that's how this whole exercise was done from something like 2008 to 2017. It was completed, and even now it is continuing. There are actually few hundred leads obtained from this in terms of novel molecules in the area of anti-cancer, anti-diabetic, anti-inflammation, and anti-infective, and that exercise still continues. I must add here that not just these 4 activities today, this repository has also been screened for variety of other activities, including production of some enzymes in biofertilizer needs, or some enzymes required for biofuel generation. So there are some success on those lines also. So the point is, such repositories are a great source of microbes for any potential applications. So this was about this. So overall, the microbes present in this repository are from diverse ecological niches. Here there are numbers of what all each institute has contributed, and the distribution of activities. Some 1,600 extracts were positive from 250,000 organisms, and they fall into different categories like anti-infective, anti-inflammatory, anti-diabetes, and anti-cancer. You will find that anti-diabetes is the largest number. As against anti-cancer, very few isolates had compounds that had anti-cancer activities. Some of the isolates had multiple activities, in the sense that same isolate producing more than 1 or at times 3 activities were produced by the same isolate. So as I said, this exercise is still going on. It is not an easy job to characterize over 200 molecules, and that also is half done. These 200 molecules have come from some 400, 500, where the entire exercise of characterization was done. Doing it for entire 1,600 is going to take a very long time, but it's going on. So again, coming back to what I started with, I talked about metagenomic approaches, and I said that from metagenomic sequence information, you can generate novel, from environmental samples, you can develop strategies to grow novel isolates and get novel bioactivities. And from metagenomics also, you can get novel activities. And when it comes to bioactives for human health, why not we look at our own body? So that is the part which I'm going to talk about in little more details, that why to go to soil, why to go to marine sediment or lake sediment if you are looking at bioactive molecules for our own body? Why not look at our own body? So that's where the metabolites, novel bioactive compounds from microbiome comes into the picture. We know that our body harbors a large number of microbes, and the estimates are anything between 5 times to 10 times more bacteria in our body than our own cells, and several thousand times more genes in our body, bacterial genes in our body, and they are found in many body sites. So nostrils, ears, oral cavity, GI tract, skin, lungs, everywhere the bacteria are found, and the total number of bacterial species in our body is more than 20,000. So it would reach something around 50,000 different bacterial species are present in our body. And there is a tremendous individual to individual variation also. So taken together, if you want to take the diversity of all human beings and microbes associated with that, I would say there are few million distinct species of bacteria exist on human body. And then that is a tremendous resource for variety of bioactive compounds for human use. And the most important part is that since these are the molecules derived from bacteria in our own body, they are not going to be highly toxic to our body. Probability is that they are not going to be very toxic to our body, and hence there are more chances of success over there. So that is the main interest why people are looking for such molecules. And I'm also going to show you an example where you don't even need to purify the molecule. You can directly use the organism that is producing the molecule can be used for the therapeutic purposes. So I'm going to show you some example of that as well. So there is a lot of talk about human microbiome. It is the biggest thing in biology after Human Genome Project. It can provide simple, sustainable, and safer solution to human health, which were not possible earlier. And it is a very powerful tool for personalized therapies when used in conjunction with human genome. So that emphasizes the importance of microbiome, and also comments from some of the well-known personalities in science, Nobel laureates, who talked about importance of microbiome. I'm not going to talk much about these things. I'm going to spend more time on main purpose of our discussion today. So the topic has also covered the front pages of many important scientific magazines. So this is something which I had already made a passing remark, that if you look at our body, the gut contains maximum number of microorganisms up to 1 kg, nose 10 kg, mouth 20 grams, nose 10 grams, mouth 20 grams, vagina 20 grams, skin 200 grams. So total weight of microbes in our body is in the range of 1.2 to 1.5 kilograms. So it is regarded as a separate organ. And this is something which I had already said, that all this interest in microbes come from the fact that only a small fraction, up to 0.1 or 0.01% can be grown in the laboratory. 99.99% remains uncultured in the laboratory. And I had also made a remark to that, people found that if you cannot grow the microbes in the laboratory, you can look at their DNA. So the DNA sequence can give us information about physiological properties about these microbes. And this was triggered in the beginning of this century by availability of variety of sequencing platforms, which started churning out sequences in billions of bases in a single run. So that triggered the explosion in this science. So the technologies also, the cost started coming down. Something human genome, something which would have cost 15,000 crore rupees at the beginning of this century and would have taken 11 years. Today, it cost just $100, and less than 24 hours, human genome sequence can be done. And as compared to human, then one human genome sequence, genome in size is equivalent to 50 to 60,000 bacterial genomes. So the same cost and same time, you can do one human genome, you can do up to 50 to 60,000 bacterial genomes. And that's why this whole metagenomics and microbiome science became very, it kind of shot up in last decade. There are many ways in which microbes can be studied. There is a taxonomic profile which does not yield much information except for taxonomy. There is a functional profiling where, or shotgun metagenomics, where you do the whole genome metagenomics. And then there are many omics. There is a transcriptomics, proteomics, metabolomics, lipidomics, glycobiomics. So it depends on which component of the biome of environment you study. There are many omics, and you can put together many omics data together to get meaningful information from the site which you are looking at. So we carry, this is already have said, we carry organisms on all parts of our body: head, nostrils, entire skin, vagina, sexual organs, GI tract, esophagus, mouth. Everywhere we carry a specific group of microbes, and each part has its own set of microbes. I'll not spend much time on this, but basically we acquire microbes from mother at the time of vaginal delivery, and then from the breast milk, and then microbiome gets stabilized at the age of 3 to 4 years. I am not going to spend much time on that. This is merely for the sake of completion. So the gut microbes play a very important role in intestinal development, they produce lot of micronutrients and short chain fatty acid. They have a very, they are metabolically very diverse. They have many other activities. They metabolize the drugs. They block colonization with pathogenic bacteria. So this blocking of colonization with pathogenic bacteria is this act is a source of novel antimicrobials, because in order to block colonization with pathogenic bacteria, one of the mechanisms they use is the production of specific compounds which inhibit the growth of these organisms, and then this becomes the source for discovering new antimicrobials. They are involved in epithelial turnover, motility, blood flow. They are involved in immune system development and maturation. So this then becomes the source for some of the bioactive molecules which act on development or blood flow or immune modulation. So this becomes the source for that. So so in short, when we talk about human health, earlier we used to talk about host genetics and lifestyle. Today we also include microbiome into that, and alteration of microbiome or dysbiosis has been shown to be associated with many diseases like obesity, diabetes, liver diseases, atherosclerosis, metabolic syndrome, and all. So again, the point to emphasize here is that since the dysbiosis is shown to be associated with disease, the one way to reduce the severity of symptoms is to use the microbes which are depleted, and that has led to the microbiome based therapies. So I have already made a mention that microbiome varies a lot amongst even individuals. So there is no surprise that across the globe the microbiome varies. And this is the point which I had made some time back, that there are three types of microbes in any organs: symbionts, commensals, and pathobionts. As long as they are in harmony, there is homeostasis. Once this harmony is disturbed, pathobionts increase and there is dysbiosis. So this could happen because of genetic factors, mutations, immune system metabolism, or microbiota modulator like antibiotics or pathogen, or environmental factors like bad dietary habits can result into increase in pathobionts and the dysbiosis and the disorder. And as I was telling you some time back, that if you want to return to this homeostasis level, then you have to push back this balance, reduce the number of pathobionts. And that is the key for microbiome therapy. There are many ways by which you can reduce the number of pathobionts. One way is just to simply purge the commensals and symbionts so that pathobiont proportion goes down. Other ways is to reduce the proportion of diet through proportion of pathobionts through diet or some other therapies. And that's all. That is the basis for new microbiome based therapies, of which I'm going to show you some examples. So this dysbiosis has been shown in diabetes, CVD, inflammatory bowel disorder, non-alcoholic fatty liver disease, obesity, many autoimmune disorders, metabolic syndrome, cancer, autism, and I'm going to give you examples for some of these. So as I said, that this paves the way for novel therapies, and then microbiome based therapies have many roles. They can help us in the prevention of the disease, diagnosis of the disease, therapeutics, and personalized nutrition. I'm not going to go into the details of that because this is not the topic for today's discussion. And there are several examples where how the personalized medicine works. And again, this particular examples, I'll revisit this if required. Right now, I'm not going to go into the details of this example where the role of microbiome in regulating blood sugar in individuals was very nicely shown. I mean, just to highlight the fact that the study shows that if you take an Indian context, that the study shows that 1 cup of rice will not lead to same level of sugar increase in everybody, depending on who that person is, how he ate the rice, when he ate the rice, with what he ate the rice, whether he had done any exercise or not, and his microbiome. All that put together will decide how much the sugar level will increase in that individual. So it is not a generalized value for everybody. It's a very, very individual specific. And that has today led to the science of what is called as Nutrigenomics or personalized medicine, individual based therapies and all. I will revisit this point sometime later. I'm not going to talk about it today. So what I want to emphasize is that we have said that these three are involved: host genetics, lifestyle, and microbiome are involved in maintaining our health. And one way to push back the balance to healthy organisms is to introduce healthy organisms from a healthy human being. So earlier people tried successfully fecal microbiota transplantation. This we are going to visit it again anyway, but just to give you basic information that in Western world there is a disease called as Clostridium difficile diarrhea, which is antibiotic resistant because it is a hospital acquired infection, and no treatment was working on it with very high mortality, more than 90% mortality. So what they started doing is that in such chronic patients, they started using fecal transplantation from healthy individual. And this therapy worked like a miracle, and they got more than 90% success. So today it is a standard therapy for Clostridium difficile diarrhea in the entire Western world. But since the fecal transplantation may not be aesthetically acceptable, there are further therapies evolving. So therapies are moving from consortia. So instead of total fecal, crude fecal transplant, you use specific consortia of organisms, then to develop a single strain, which is very effective, and finally to arrive at a bioactive molecule from this strain, which is actually responsible for the success of the organism. So the microbiome based therapies are now rapidly moving from crude fecal microbiota transplantation to bioactive molecules. So they are becoming more and more specific. So, and I'm going to show you some examples of that to make you believe what I'm saying. So the microbiome based therapies, as I said, they fall into multiple categories: fecal microbiota transplantation, microbial consortia, single isolates. Today there are some efforts to use genetically engineered microbes also, but eventually it's a long way when such genetically engineered microbes would be permitted to be used in actually human beings. So this possibility exists, but we are not going to talk about that. And then the bioactive molecules. So these are the many ways in which microbiome based therapies work. And Clostridium difficile diarrhea, I have already given you the example that it showed more than 90% success. And people are also trying to develop capsules or consortia of microbes for treatment of Clostridium difficile diarrhea. So umpteen number of pharmaceuticals have come up with umpteen number of consortia, and these consortia are in different phases of clinical trials. You would know that anything that is to be used in human beings has to go through multiple stages of experimentation in human, limited trials in human volunteers. I mean, you must have heard all these things more closely when you talked about development of vaccines for corona. You heard about first phase trial, second phase trial, third phase trial. So anything that is used in human being has to go through all these trials. So these microbial consortia are in different stages of clinical trials. I'm not going to read out all these things. I'm showing this slide just to tell you the sheer number of success stories of using microbial consortia for the treatment in humans. So the fact that so many such studies are being done means the therapy has a potential and it is being used. So there are many such studies like Crohn's disease, Clostridium difficile diarrhea, ulcerative colitis, Crohn's disease, Crohn disease. Many such consortia are in different phases: discovery, that is first phase, phase 1 clinical trial, or phase 2 and 3 clinical trials. I'll just quickly run through such different trials. I'll not describe each and every trial because it is irrelevant. So there is one example where single strain with live biotherapeutic product. This drug is Blotix. It is available for irritable bowel syndrome, and this is under phase 1 clinical trial now. So the fact which I was telling you, that the science has moved from microbial consortia to single strain and single strain live biotherapeutic product. This is the example. So this particular product is under phase 1 clinical trial, means it is not just a research concept, it is actually going to come into the practice. So like that, there are many such trials are going on as I said that, and then this is the example of small molecules or microbe derived metabolites. You can see here, this is Fimbri adhesin inhibitor. It is again phase 1 clinical trial for Crohn's disease. One such molecules, SYN004 for Clostridium difficile diarrhea, is under advanced phase 2 or phase 3 clinical trial. Another molecule for inflammatory bowel disease is at the research stage. So you can see what I said earlier was crude FMT to consortia to single organism to actual single molecule derived from a gut microbiome. You can see it actually happening. You saw the earlier examples where people use consortia. Then from consortia, there was a single strain, and from single strain, now you have small molecules derived from components of gut microbiome. So that is actually happening. This is the 2020 review, and this is again a summary of status of different microbiome based drugs, which is in under which phase 1, phase 2, phase 3 clinical trial, a summarized information of that. And the first product to actually come in the market very soon is going to be this SCR-109 orally administrable microbial consortium for recurrent Clostridium difficile infection, because it has passed phase 3 clinical trial recently, and this is the publication which has come in January 22. So soon this product will be there in the market. Of course, not in India, but in the Western world, this product will be in the market. But this is a consortium. You have also seen examples of actual molecules being in the phase 3 clinical trial. So they would also soon be in the market. So there are various approaches by which one can look for bioactive compounds in microbiome. One can do a comparison of germ-free mice and healthy mice, look at the metabolite pattern and identify the molecules. You can actually do a specific therapy, like you can do the comparison with diabetic and healthy, and then from healthy you can pick up the molecules which are responsible for suppressing the diabetes. So many such approaches can be used for metabolite screening and actual isolation of organism. You can, this I have already talked to you about genome mining. So you look at the metagenome, do predictions of pathways, find out the putative structural molecules, and then generate those metabolites and use them for actual treatment. So there are many such approaches that can be used, and this is just to show you the diversity of metabolites that are produced by gut microbiome. So these are ribosyl synthesized post-translationally modified peptides like lactosilin. Then there are mycolic acid, galactosyramide, indole propionic acid. These are the products of amino acid metabolism, indole propionic acid or tryptamine. Then there are non-ribosomal peptides like corinibactin. Then there are polyketides, there are terpenoids, there are oligosaccharides. So a wide range of metabolites are produced by gut microbiome, and all of these have a potential as a bioactive molecules for therapy. And this is this is just not not to scare you. Even if this is unreadable, don't worry about that. This is just to show you that what is the diverse range of microbially produced metabolites that are currently known and have variety of activities against different pathogens. So these are products of amino acid metabolism, oligosaccharides, glycopeptide, terpenoid, non-ribosomally produced peptides, polyketides, porphyrins, ribosomally produced peptides. They all have certain molecules produced and which have activity against variety of pathogenic bacteria. So this is a tremendous source for novel bioactive molecules. And so this is, again, I'm going to just list out some of the compounds of which are known to be produced, antimicrobial peptides known to be produced by actual member of our body microbiome. So from nasal microbiome, you have staph and staphylococcus staph producing these two molecules, bacteriocins, which are active against gram-positive strains and staph aureus and gram-vancomycin resistant enterococci. Then you have from oral microbiome, again you have different organisms producing variety of peptides which are active against indicator strain pathogens. Then you have gut microbiome, largest reservoir of antimicrobials. These are all characterized, known, shown, published molecules. Every place I have shown the references. So these are all well characterized molecules produced by members of the microbiome, skin microbiome, vaginal microbiome. So a large number of members of microbiome are known to be producing biologically active compounds which are of clinical significance. It is not just anti-infectives, but they are known to produce some anti-cancer drugs also. And this is one example, Staphylococcus epidermis. It produces a compound 6N-hydroxyl aminopurine, 6-HAP, which is known to have anti-cancer activity, and it is effective against skin melanomas. So the purified compound has also shown the activity against the melanomas in experimental mice. It has shown activity in cell culture also. But what these people did was, instead of using purified compound, they use the organism that is producing it. Instead of having the chemical, they use the organism as a member of skin microbiome experimentally, and they found that even the organism is effective as the purified compound. So this is a kind of new way of treatment of such diseases. Instead of having a pure drug, you can use kind of microorganism based therapies, which is successful. So there lies the importance of microbiome based therapies. So there are again many such compounds which are in different phases of clinical trial. And with this, I think I have completed my time. Thank you very much for your attention. And if there are any questions, I'll be more than happy to answer. Thank you.
Thank you, sir. Now this topic is open for discussion.
Hello, sir.
Yes.
Hello. Thank you, sir. Actually, your presentation was very informative. Also, you have mentioned regarding the microbiome of human body. Actually, they play very important. Yes, but
Dinesh,
it may be correct or wrong. I don't know. Can we use microorganisms or microbial activity or microbial products for detection of cancer, early detection of cancer or other detection of cardiovascular diseases?
That's a very good question. About many diseases, including cancers like colorectal cancer, people have done studies in which, for example, in diabetes or in colorectal cancer, this is established that as the individual progresses towards the disease stage, the microbiome changes. And that's how the microbiome alteration in microbiome can be used for diagnostic purpose. So in future, such tests are possible for colorectal cancer or maybe for diabetes and other diseases. But what I can tell you is that one particular disease known as non-alcoholic fatty liver disease. So as the name suggests, it is the disease of the liver. And for that disease, the treatment was, the diagnostic test was liver biopsy. But today a test has been patented wherein the diagnosis can be done based on fecal microbiome. So now here you see the example where a invasive liver biopsy is replaced by microbiome based tests. So indeed, in future, it is possible to develop microbiome based tests or doing a early diagnosis of diseases, disorders like diabetes or CVD. It will be possible in future. Yes.
Actually, main present day problem is the cardiovascular diseases and cancer.
Right.
The diagnosis late stage, but for
Right.
the early stage, it's very beneficial to the both community, doctor community as well as patients also. Okay.
Right. So the research is going on in this direction, and see, it is again has to be since microbiome is different in different populations, unless you have a specific causative organism like your gastric cancer and H. pylori. There is a single organism associated in all the cases. Here, the microbiome being region specific, you have to do a study on local population. So for India, we have to generate the data on how microbiome changes, for example, in diabetes or in colorectal cancer or CVD in the Indian population, and then develop the diagnostic markers.
Thank you, sir. Thank you.
Thank you. There is one, Dinesh Zadav, he has a question. Dinesh, you can either ask question or you can type it in the chat box.
Hi, Professor. Can you hear me?
Sure, yeah, I can hear you.
Yeah, so it was a good talk. I just had one question from the end of your talk. At the end, you talked about that hydroxylamine purine is showing anti-cancerous activity. So I was just wondering like how it is inhibiting cancer and what exactly it's doing at the molecular level.
As you know, I mean, as the structure suggests that it is a base analog. So it works like a standard base analog wherein it gets incorporated in the DNA at the time of DNA replication and then inhibits the further DNA replication. So it functions like any other base analog. There are many other base analogs are also being used for therapeutic purposes. So it works that. But if you want a detailed answer, you can drop me a mail. You can find my email ID easily on the net. It is yogesh@gmail.com or yogesh@nccs.res.in. I'll give you a detailed answer.
Okay. I was just wondering if it is inhibiting any protein which is involved in
So
metabolism.
we'll talk about that. You can just drop me a mail. I'll share some more information with you.
Yeah, sure. Thank you. Thank you so much.
We do not have any capacity to abstract this talk. However, we truly inspired with this. I just can say thank you to Honorable Yogesh Sauchi sir for sharing huge information, especially for giving applications of dynamic human microbiome based therapies also. Sir, we also wish to declare you guest of honor for conference, and on behalf of organizing committee, I request you please accept this.
Thank you. My pleasure and honor.
Thank you, sir. Thank you.
Different.
Thank you, everyone. Moving towards next talk, I would like to invite Dr. Amarja Zoshimam, who joined us virtually, and ma'am is from also Ncmr Pune. So I would like to request Dr. Hemalata Bhosle ma'am to please introduce Amarja Zoshimam. Ma'am, please.
Good afternoon. Women's empowerment is a process in which women gain greater share of control over resource material, human and intellectual like knowledge, information, ideas, and financial resources like money and access to money, and control over decision making in the home, community, society, nation, and to gain power. Since morning, we are listening and watching the research contributions of male scientists. Okay, in this session, we have two women representatives. Okay, Dr. Amarja Zoshimam and Professor Anita Patil ma'am. I feel very proud and happy to introduce today's first woman scientist, Dr. Amarja Zoshimam. She has completed her PhD from Agarkar Research Institute, Pune, under Savitribai Phule, Pune University, Pune. She has research experience of about 12 years in the field of microbial taxonomy, systemics, and ecology of microorganisms, isolated from various extreme environments with representatives of Lonar Lake and Pangong Lake. She is currently working as Scientist C at National Center of Microbial Resource, Ncmr, NCCS, Pune, Maharashtra. She is also in charge for General Deposit Service for Phylum Firmicutes and Curator for Phenotypic Characterization Service at Ncmr. She has 14 research publications that are published in very reputed national and international journals. Many more are there, but some representative journals are there. Just like Pathak Madam said that some journals are dream journals and everyone wants to publish their research articles in that. So some representative dream journals like Microbial Ecology, Annals of Microbiology, International Journal of Systematic and Evolutionary Microbiology with 5 book chapters. She has guided more than 12 master dissertation theses, and she has attended and organized many national and international conferences and workshops. With this brief introduction, I request Dr. Amarja Zoshimam to deliver her talk.
Am I audible? Hello?
Yes, ma'am. Yes, ma'am.
yes,
You are
ma'am.
audible.
Okay,
Yes,
okay.
please.
Good afternoon, everyone. Thank you so much, ma'am, for your kind introduction. As she said that from morning we are listening very nice lectures and very from various eminent scientists, and this conference gives me a chance to listen Dr. Yogesh Shauche's lecture after a long time. So for that purpose also, I am very thankful to you. I would take this opportunity to thank Dr. Anupama Pathak Madam and Director SRTM University, Nanded, for giving me this opportunity to involve in this international conference and give my talk in front of you. So I will start with my topic. Today's topic is alkaliphilic bacteria from Lonar Lake, a potential source of antibiotics. As already Madam told that most of my work, or I am mostly interested in studying taxonomy, systematics, ecology of extremophiles. So my PhD is also from various alkaliphilic bacteria from Lonar Lake. Then I have studied various bacteria from Pangong Lake, as well as some other extreme ecosystems. We have isolated the organisms and studied bioactive compounds from those microbes. So today the topic is from antimicrobials from Lonar Lake. So we will start with what is an antimicrobial compound. So these are secondary metabolites, and which are defined as low molecular weight organic natural products which are produced by microorganisms and active against other microorganisms at very low concentration. There are two types, as they may be microbistatic, that is they inhibit the growth of other microorganisms, or they are microbicidal, that is they kill other organisms. There are various sources such as natural and synthetic, and why they are produced to survive in the environment and compete with other microorganisms for resources. From morning, we are hearing there are n number of antibiotics are produced and marketed, but still, why there is a need of novel antibiotic? So we all know day by day, due to our lifestyle, due to pollution, due to other environmental stresses, various new illnesses are coming. To overcome these new illnesses, we are making a lot of antibiotics. Due to use and misuse of these antibiotics, new class of organism is emerging day by day, which is termed as antimicrobial resistant bacteria, that is AMR bacteria. These AMR bacteria, which are also known as MDR bacteria, and thus to overcome all these MDR bacteria or to deal with all these MDR pathogens, there is a need to develop novel antibiotic. So why extremophiles? As we know that extremophilic bacteria follow different metabolic pathway than normal organisms, which lead to the formation of variety of secondary metabolites, which include antibiotics, growth promoters, enzyme inhibitors, and so on. In recent years, they have been recognized as valuable sources of novel bioproducts, and this may well include antimicrobials. Little information is available on the production of antimicrobial compounds from these extremophiles, and thus it is worthwhile to study these bacteria. Here we study alkalophiles isolated from Lonar Lake. All of you know that Lonar Lake is an Indian soda lake with pH of 9.8 and NaCl concentration of 1%, which is situated at Buldhana district of Maharashtra state in India. So based on this background, we prepare our objectives of our study as screening of these alkalophilic bacteria which are isolated from Lonar Lake for production of antibacterial compounds, complete characterization of selected isolates using polyphasic approach, extraction of these compounds using various solvents, and optimization of culture conditions for production of these antibacterial compounds. The main important step here is which pathogens are to be included in our study. So we started our study way back, and at that time, WHO published a list of MDR pathogens. We have selected a few pathogens which are mainly causing the infections in hospitals, and they are pathogenic to various patients, to ventilators as well as catheters. And so there is alarm from the WHO, and there is a need to develop or need to take immediate step and develop a novel antibiotic against all these pathogenic bacteria which are MDR in their nature. And so we have selected these 4 pathogens in our study. These are Escherichia coli, Acinetobacter baumannii, Klebsiella pneumoniae, and Staphylococcus aureus. So Escherichia coli, you all know 3 of them are gram negative in nature and 1 is gram positive. Escherichia coli, you know, causes urinary tract infection and diarrhea. Acinetobacter baumannii causes bacteremia and various wound infections. Klebsiella pneumoniae causes respiratory tract and surgical wound infections, and Staphylococcus aureus mainly causes skin diseases. So all these pathogenic cultures were procured from Backtest Lab, Nashik, and once we procured them, we have tested their authenticity using 16S rRNA gene sequencing at Ncmr and deposited at our culture collection with MCC numbers. once we check the authenticity, we have to check MDR pattern of all those pathogens. So to check the MDR pattern of these pathogenic cultures, antibiogram of these pathogens was obtained using standard method. And here you can see that all of these pathogenic cultures showed resistant to most of the antibiotics under study. This is the zone interpretation of these pathogenic cultures towards the antibiotics. And here you can see that all of these pathogenic cultures are resistant to most of the antibiotics. Then the first step is to screen these isolates for their antibacterial activity. So first step is primary screening of these isolates for to study their antibacterial activity. Here, spot on lawn method is used. We have isolated total 210 alkaliphilic bacteria from Lonar Lake, which were spot inoculated on alkaline nutrient agar. In all these experimentation, we use alkaline pH as the pH of Lonar Lake water is around 9.8 to 10. And incubated at 37 degrees Celsius for 70 to 96 hours. After this incubation, we took 18-hour-old fresh cultures of indicator pathogens. As we have already discussed, these 4 pathogens were taken, which were mixed in MH medium, which is of pH 7, and which are overlaid on grown Lonar cultures. These plates were then incubated at 37 degrees Celsius for 48 hours and which were further observed for inhibition zone of inhibition. Here, tetracycline and streptomycin discs were used as positive and negative control. This is how the plates looked after 48 hours. This is the results of primary screening, where you can see big zones of inhibition around the culture. And so out of 210 Lonar Lake isolates, 44 isolates shows antibacterial activity against 1 or more indicator pathogens. So this is the very, very promising results. So this is the table where you can see that these are the various strain IDs which have been isolated from Lonar Lake, and most of them showed antibacterial activity against more than 2 MDR pathogens. So from this table, we have selected these 2 isolates for further study due to the as they show positive results, or they show zone of inhibition for all the 4 pathogens. So both the isolates were further subjected to secondary screening, where the 2 isolates were inoculated in 100 ml of alkaline nutrient broth under Sheck culture condition at 37 degrees Celsius for 96 hours. The broths were then centrifuged for 30 minutes and supernatants were then subjected to membrane filter sterilization. 50 microliter of filtered supernatant was then subjected to agar well assay method. So these are the plates showing zone of inhibitions, and here this is the results of secondary screening, where you can see that MEB 199 showed zone of inhibition against E. coli as well as Acinetobacter baumannii, but no zone of inhibition was observed in case of MEB 071, though the culture showed big zone of inhibition in the primary screening. So I will discuss this point later. Once we are finished with primary and secondary screening, the next important step is to identify these 2 selected isolates. So the identification is a very, very important step. And first we have started with optimizing the pH required for growth of these 2 isolates. And you can see from these graphs as they both the cultures optimally grow at pH 10. Then, as I already told that we have carried out complete characterization of those isolates, and we have started with morphological, physiological, biochemical characterization. So here you can see the gram staining photographs of these two isolates, and here MEB 199 showed long thin gram positive rods, while MEB 071 is a gram negative thin rod. So MEB 199 was further subjected to spore staining, where you can see central oval spore in their cells. So this is the complete table of the characterization of these two isolates, and where you can see the complete characterization was done from morphological up to, then chemotaxonomic and molecular. All the complete characterization was done using Ncmr facility, and here you can see that for a strain MEB 199, complete characterization was now over. And so I will go with that culture. That is, the culture produces irregular, colorless, and opaque dry colonies on nutrient agar, alkaline nutrient agar medium. And the optimum pH for growth is 10, with tolerance up to 0 to 10%. And it has these are the various fatty acids present in the cell membrane of this organism, and these are various polar lipids present in the cell membrane. Based on 16S rRNA gene sequencing, the organism showed sequence similarity of 98.36% with alkali halobacillus alkali nitrilicus. So the organism was further subjected to whole genome sequencing, and which showed the similar result. Okay, so this is the phylogenomic tree, and this is the UBCG based tree, which is constructed using 92 bacterial core gene sequences, showing the relationship of members of genus Alkali halobacillus and nearest genera. So here you can see that Lonar Lake strain, that is MEB 199, showed close resemblance with Alkali halobacillus alkali nitrilicus, and it shows only 98% of similarity. And here I have highlighted this culture also. This is a type species of this genus. This is Alkalohalobacillus alkalophilus. And our strain, that is MEB 199, showed only 94% sequence similarity with this type species, and which indicate that it is distantly related to genus Alkali halobacillus. So it means that it now belongs to some different genus than Alkali halobacillus. As we have studied genome of this culture, based on that, we determine different values such as G+C content of genomic DNA, which was 36.4%, digital DDH value, ANI value, and these values between our strain and closest relative confirm the novelty of this new species. So I have already discussed that our strain is novel genus, and these values also confirm that it is not only novel genus, but it is novel species as well. So based on polyphysic taxonomic analysis, the strain represents novel species of newly proposed genus, for which the name Alkali Halobacterium elongatum genus Nova, species Nova, is proposed. And the work is published very recently in Frontiers of Microbiology. As the genome of MEB 199 was studied, anti-smash bacterial version 6 was used to understand the secondary metabolite biosynthetic gene cluster in this strain. And this analysis also showed that the strain has 12 secondary metabolite biosynthetic gene clusters for synthesis of various secondary metabolites such as siderophore, terpene, carotenoids, ectoin, and so on. So this also confirmed that it is producing some bioactive compound. Okay, so this is all about detailed study of strain MEB 199. Again, coming back to the previous point that MEB 71 produces big zone of inhibition in the primary screening, but when the culture was grown in the broth, The broth was centrifuged, and when the supernatant was put in the supernatant was then assayed using well assay method, it didn't show any zone of inhibition. You can see in these plates. So then we have decided to study cellular distribution of antibacterial compound in this culture. For this purpose, MEB 71 was inoculated in 100 ml of alkaline nutrient broth, and the culture was kept under shake culture condition at 37 degrees Celsius for 96 hours. Culture broths were centrifuged at 3800 G and for 30 minutes. Here we would like to check whether the antibacterial compound produced by MEB 071 is extracellular, intracellular, or cell bound. So for that purpose, what we did, we took supernatant and subjected to the agar-well assay to check whether it is extracellular. Secondly, we directly we centrifuged the culture, take the cell pellet, which was subjected to the osmotic shock and sonication, further subjected to centrifugation, and then intracellular material was subjected to agar-well assay to check whether the antibacterial compound is intracellular. And thirdly, culture was directly spotted using spot on lawn method to check whether it is a cell bound. Okay, so here these are the results, and here also we can see that here again the same result. Here intracellular, there were no zones. Cell bound, again there are zones you can see here, and in extracellular, where the supernatant was added, but you can see the culture was grown here. Yeah, so there is also a zone. So from all these observations, we conclude that our product or our antibacterial compound, which was produced by MEB 71, was extracellular, and it may not come in the supernatant when we centrifuge that supernatant, or we have to study in depth the growth curve of the organism, and at that stage only we will remove the culture supernatant and again check its activity. Okay, so to once we get the antibacterial compound, the next step is to concentrate and again check the activity. And here two different methods were used for concentration. First is solvent extraction, where cell broth of MEB 199 and cell pellet of MEB 071 were used. And these three solvents were used for solvent extraction: chloroform, butanol, ethyl acetate. And here we observed that MEB 071 can be extracted using ethyl acetate, while MEB 199 can be extracted using chloroform. And this is the lyophilizer at Ncmr. Here also we use this lyophilizer to concentrate the supernatant and again subjected to agar-well assay, but we didn't get good results in this after lyophilization. Okay, so this means that lyophilization affect the process or affect the product structure, and so that it didn't give any zone of inhibition. Next step is optimizing the culture conditions for production of this antibacterial compound. And here we use various cultural conditions such as incubation period, pH, NaCl concentration. Incubation period from 24 to 96 hours was used, then pH from 7 to 11, and NaCl concentration from 0 to 10% were used to optimize the bacterial antibacterial compound production. Here both the cultures were inoculated in nutrient broth with respective parameters, subjected for centrifugation, And here, MEB 199, again, supernatant was taken and subjected for agar-well assay. And for MEB 071, cell pellet was taken and using spot on lawn method, the assay was carried out. So these are the results. And here one can see that for MEB 071, this is it produces optimum antibacterial compound after 72 hours. And for MEB 199 could produces this compound after 48 hours. Then optimization of pH, that is here also both the cultures could produce optimum in compound or optimum antibacterial compound at pH 10. And this is the graphical representation where which showed that both the cultures could produce optimum or maximum zone without NaCl. So here you can see that both the cultures have producing big zone when there is no NaCl in the culture medium. Okay, so coming to the discussion here, I would like to say that we have isolated around 210 alkaliphilic bacteria. Among that, 44 are positive, and we have identified all the 44 bacterial cultures using 16S rRNA sequencing. And these are few representative bacterial cultures or genera among those 44. And here you can see that there are a lot of bacterial strains such as Halomonas, then Pseudomonas, Sabulonegiri, Rosinatronobacter, Monicus, Nitrincola, Laccisaponensis. This organism was already published as a novel in IJCM. Then Alkali Halobacterium elongatum, which was recently published. Then Providentia, Alkalimonas amylolytica, Rinmera perlucida, and Alishvenia geotali. So these are various different genera with their species and showing the zone of inhibition against 2 or more MDR pathogens. Okay, so this is the comparative study which was made with bacterial cultures which have been isolated and reported for antibacterial activity from Lonar Lake, and which we have isolated and characterized at Ncmr. So this table clearly indicated that the cultures which we have isolated, all the cultures, first thing is all are alkaliphilic in nature. They grow only at 9 to 10.5 pH. All are all showing sequence similarity with obligate or alkaliphilic bacterial cultures. And here you can see that these are different from reported ones. And very important thing is most, or I will say that 50% of these bacterial cultures are novel. The study is going on, and we will definitely publish those novel cultures. So in conclusion, I would like to say that through this study, we obtain or we isolate 2 taxonomically important alkaliphilic bacterial strains, that is MEB 071 and MEB 199, producing antibacterial compound against 4 MDR pathogens at pH 10, which were isolated as well as characterized. And 44 strains are showing antibacterial activity, and most of them are likely to be novel. Here I would like to make my important point as they are likely to be novel, and thus antibacterial compound produced by them may also have different properties or structure. Antibacterial compound which we got from or which we have isolated from these 2 cultures, the structure and characterization is under process. So from the findings of this study, it can be concluded that alkaliphilic bacteria from Lonar Lake should be taken into the consideration in search of novel antibacterial metabolites. So this is all about our work which we have done at Ncmr. I would take this opportunity to highlight few points regarding NCMR. Already Dr. Yogesh has showed or discussed, or he has highlighted many things about our institution, but these are few things I would like to tell about our institute. That is, this is National Center for Microbial Resource. This is the single largest culture collection in the world with more than 1.4 lakh microorganisms, which include archaea, bacteria, fungi, and yeast. And major part of its collection is from diverse ecological niches, as Yogesh sir has already shown you a slide about this, and which make this collection a unique one. And it played a vital role in exploration and conservation of microbial diversity of India. So our main objectives are to serve as leading world-class microbial resource repository, provide authentic, high-quality services for microbial preservation, characterization, authentication, and supply to industry and academic institution. As I already, as mentioned here, characterization. So you can get a complete characterization of your novel strain at our center. You can preserve your precious strains for long term at Ncmr. So you can get lot of services at our center. And to conduct research in the fields of microbial economy, taxonomy, and systematics. And fourth important point is to serve as an IDA and under Budapest Treaty and designated national repository under Ministry of Environment and Forest. Under IDA, we accept patent as well as safe deposits for long-term preservation. Okay, so this is the diagrammatic representation which services Ncmr is offering currently. Our main service is culture deposition, under which we accept general deposit, patent, safe, as well as AMR culture. This is the new addition to the deposit, that is antimicrobial resistant cultures we are accepting as a deposit. Second most important thing is identification or characterization. This is the very important service, as this service provides a complete profile of your organism, starting from morphology, physiology, chemotaxonomy, and molecular characterization. So here you can get various services under morphological and biochemical characterization. We provide services like API, Biolog, Vitek, and so on. Then under chemotaxonomic characterization, we provide services by using FAME. Then we are doing polar lipids, and using MALDI-TOF, we will provide protein profile and identify your culture within very short time. And third is molecular characterization using 16S rRNA gene sequencing. Then Ncmr is also providing services in the fields of genomics, where you can get your whole genome sequencing, and related bioinformatics studies also you can get under this service. And culture supply, we supply the authentic cultures to various industries and academics, and various customized services like isolation of DNA, purification of mixed cultures, then multi-profiling of your strains, and so on. So this is my kind request to all of you to kindly visit our Ncmr website and deposit your strains, take our services, and benefit yourself. And you can visit our center at any time. Thank you so much. And this is our team. Any work in at this center is not completed without a team. So this is a teamwork. And finally, I would like to acknowledge DBT Government of India for funding this facility. Director NCCS Pune, Dr. Avinash Sharma, in charge Ncmr, NCCS Pune, Director School of Life Sciences, SRTM University Nanded, and Dr. Anupama Pathak ma'am for giving me this opportunity to be a part of this beautiful conference. And Lastly, I would like to acknowledge all the Ncmr staff for their help and support. Thank you so much. Hello.
If you have any questions. No questions? Thank you, ma'am, for this informative talk and giving in detailed information. Means any new student who has entered in the field of research, how to isolate bacteria, how to screen them for bioactive molecule production, and how to characterize them. Detailed information and basic information that is given by you. Particularly the bioactive molecule producing bacteria, how they are isolated from extremophilic environments like lunar lake.
Yeah.
This aspect is covered by you, ma'am, in very detail. This will be definitely helpful to our students and researchers working in the area of extremophile or extremophilic microorganism. Also, I'm thankful to you for giving idea about what kind of services are offered by NCMR and NCCS. And we expect similar assistance in future also. And expect your next talk in person in life sciences or SRTM University. Thank you, ma'am.
Thank you. Thank you so much, ma'am. Yeah.
Thank you, ma'am. Thank you very much for your nice and informative talk, ma'am. Now, moving towards next technical session, I would like to welcome Professor Anita Patil, ma'am, who joined us with online from Santa Gadge Baba University, Amravati. Ma'am, are you there? Anita Patil, ma'am. Am I audible, ma'am?
Yes, ma'am, I'm there. Yes.
Now, I would like to request Professor
Welcome, ma'am. Dr. Anita Patil, ma'am, she is a professor in the Department of Biotechnology, Sant Gadge Baba, Amravati University, Amravati. She has completed her PhD in biological degradation of lignin during biopulping of wood. Her research interests include plant and microbial secondary metabolites, their production and biological activities. And under various abiotic and biotic stresses, whatever plants and microbes are growing, that are considered as her research interests. She has focused on identification of newer bioactive molecules, particularly from therapeutic plants and endophytes from tribal areas. She has established a multidisciplinary and nationally recognized comprehensive research in the area of biological properties of various therapeutic plants and microbial purified extracts as antimicrobial, anti-urolytic, anti-hemolytic, and antioxidant properties. She has developed a significant commercial product for common urinary lithiatic disorder from therapeutic plants, and this product is commercially to be launched by Nagpur MIDC and Nagpur MIDC based company Dr. S. Kumar's manufacturers, Appropriate Diet Therapy Center and Rajiv Gandhi Science and Technology, Government of Maharashtra, and the Sant Gadge Baba Amravati University, her own institute. She has also experience on working anti-malarial drugs and the drugs which are active against phytopathogens by trichoderma, especially their secondary metabolites. She also did her collaborative research with Toshio Tajima from Tokyo, Japan, particularly for organic farming at Kaji Manidara village for organic rice growth as promotional activity of PGPR. She has authored and co-authored over 80 peer-reviewed research papers in journals of national and international repute. She has written several book chapters, proceedings, and popular articles to her credit. She has authored and edited 3 books. She has obtained 3 patents. 2 are in the final stage of granted, and 2 new patents are filed by her. She has completed several research projects funded by different funding agencies like UGC, DST, RGSDC, BRNS, DAE, etc. She has also participated in various national and international conferences, workshops, and conducted various trainings for teachers and students. With this brief introduction, I welcome Patil ma'am to deliver her talk.
Thank you, madam, for your kind introduction to me. And first of all, let me thanks to organizer for giving me this opportunity to speak on in this conference. So mostly I was thinking that the research students are the part of this conference, and they need to understand how to start with the research with the medicinal plants. So always to remember that impact. This is the word that is called as Indian medicinal plants, phytochemistry and therapeutics. Now this is a curative database which has the information on 3 types of association of Indian medicinal plants, phytochemical composition and therapeutic applications. Whatever the plants you are going to identify, you are working on that plant information has been gathered in a particular database, and that is the impact. So always remember, actually my talk is on artemisinin. That is a terpenoid, and this was a topic, no, artemisinin. And whenever you wanted to go for research of any plants, the plant selection is very important. And the selection of the plant has to be always done by the traditional approach, the literatures, the folk claims, the bulk screening, and the phytochemistry. Along with this, in the modern sciences, you need to have a modern approach for identification of the plant. Now, you know that the plants are the machineries producing some phytochemicals. You know, they are the different types of phytochemicals: alkaloids, anthocyanins, flavonoids, lignans, quinins, steroids, and terpenoids. Now, these are the secondary metabolites which can be exploited as a future drug or the candidate for the drugs. You know that in a plant, you need about 6 to 7 years for the discovery of a drug, because from the drug target, you have to go up to the clinical trials for the identification of a lead molecule and up to the percolation to the society. Now, if you want to discover the drug from the plant, you know that plant selection is very, very important. Its preliminary studies are important. How to go for its extraction, the isolation, the characterization, the phytochemical evaluation, the drug development, maybe of which type, the local, systematic, or oral, and the final thought that is always to be given for the drug discovery. You know that extraction of the plant drug is very, very important. From the ancient time, we are using plant in Ayurveda, and that plants are extracted by the conventional techniques or the ancient techniques. And now in a modern era, we have given, along with the traditional one, we have given a rich in techniques that has been used for the extraction of the drug. After the drug extraction, you know, isolation, purification, separation, and characterization of the drug is very, very important. From the common lab techniques to 3D, 4D, Malditov, these techniques can be used for the identification of the drugs. So any drug, if you are isolating from the plant, from the microorganism, or any secondary molecules, that activity has to be always taken into consideration in the form of pharmacological activities, the biological activities, the microbial activities, and the toxicology. Always remember, any drug that has to be assessed by its toxicology. There are the various methods, in vitro, ex vivo, and in vivo, for the therapeutic evaluation of the drug. Now, my main concern is terpenoids. We know that there are the different types of 7-8 class of molecules that has been produced by the plants. And there are, if we see, there are the 3 broad categories of the plant secondary metabolites as a natural products. Number 1 is terpenes and terpenoids. So there are about 25,000 different types of terpenoid molecules. There are the alkaloid molecules, there are about 12,000 molecules, and the phenolic compounds, these are about 8,000 types. So out of that, the terpenes, if you see the diagrams of the pie chart, if you see this, the terpenes are the most numerous and structurally diverse plant natural products. And therefore, I can feel that, I can state that the terpenoids or the terpenes are the most important drug molecules or the candidate for the, as a potential for their in industrial and the medicinal applications. If you see the terpenes, if you see the class of the terpenes can be classified into monoterpenes, sesquiterpene, diterpenes, sesquiterpene, tioterpenes, and tetraterpenes. And this is totally dependent upon the number of carbon atoms as well as the isoprene ring, which is the basis of the phenols or terpenes. So if the rings are, if the 2 rings are there, then it is a monoterpene. So according to the number of isoprene unit, the name has been changed out. So it is very amazing to see what are the different Asian techniques for isolation of terpenoids. If you see the literature, the terpenoids are being the most important biomolecules from the Asian time to be used as a drug. So in Asian times, the first time these terpenoids are isolated by the low temperature chromatography. Later on, it is refluxed the leaves in ethanol and then dissolving into water solvents by the column chromatography or by the liquid-liquid extraction. Nowadays, 2-dimensional gas chromatography, time of Fright mass spectroscopy has been used to analyze volatile oil in the leaves and the flowers of Artemisia annua. So you are amazing to know that the typical class of these molecules do possess different characteristics. And mostly the plants which are flavored one, those have some smell, they do have some phylogenetic resemblance to the synthesis of the terpenes. Now these terpenes are mainly associated with the coniferous, but nowadays there are some are found in angiosperms also. Their highest concentration found in hardwood and the lowest in the sapwood. And these terpenes are elicited via abiotic or biotic stresses. They are formed in plants in response to the herbivory or a stress. The flowers, mostly these plants do have flowers, and they used to emit the terpenoid to attract the insects as a method, and also a plant defense also. They act as in a soil or in a nearby area. They will act as a chemical messenger, which will influence the expression of the defense gene of as well as self, as well as the neighboring plant. So these terpenoids are very important molecules in nature also. If you are amazing to know that these do have some environmental aspect also. Terpenoids do have environmental aspects also. There is a global emission of isoprenes in a 500 tetragram, and they do have some interaction with the solar reactions. For example, the biogenic volatiles like alpha pyrin and alpha limonene, they do have some effect on ozone formation, because where in the Mediterranean area where the citrus is grown, they do have some effect on the ozone layer. It has been found that large majority of the terpenoid, they are released from some plants and they are chemically oxidized. Otherwise, they are transformed into different aerosol compounds in nature. Means the smell of the soil, the smell of after the water comes down, they do have some interaction with that. Stomatal conduction is very important in case of when the plants interact with the abiotic atmospheric conditions such as temperature, water availability, and irradiation. In that case, these terpenes are released from the plants, and they do have some smell and the fragrance. So this is the importance of the terpenoids. And in generally in society, we do have lot of advantages of the terpenoids. They are antibacterial, mostly anti-malarial, biotoxic effect. They are used for bioremediation. Also, there are some, along with that, they do have some effect like a causative agents for contact dermatitis, and they do have some fragrance allergy. Some of the people may have faced this problem when they go for a morning walk. This type of things can be often. So my topic was specifically on artemisinin. Now this is a cis-terpene lactone molecule, means it is a typical terpenoid, and this is artemisinin is specifically used against as an anti-malarial drug from Artemisia annua. We know that in India, or first we will say in malaria in the worldwide, and it is very problematic conditions worldwide, and there are about 300 to 500 million clinical cases, and between 1.5 to 2.7 million deaths. Out of that, only 90% occurs in Africa. In case of India, if you see that our most of the population lives in a high transmission area, and even in India, we do have malarial, still we do have malaria and cerebral malaria cases. So if we see the top 10 states in India for malarial cases, our Maharashtra stands at 6th position. So if you see this artemisinin, now this has been this is a typical molecule metabolite which is found in Artemisia annua, and this is a common plant that is being used in a traditional Chinese medicine, and it is this artemisinin contained in Artemisia annua is very low, actually 0.01 to 4.4%, and it has been reported by my co-worker, my collaborator from the bar, Dr. Fujale, that its production can be increases up to 1%. And if you provide, if the artemisinin production is from artemisinic acid, so it can increase the tenfold. So this Artemisia annua, that is its Chinese name is Pingho, is only known as a source of cis-terpene lactone. It's called as wing shu, wherein endoperoxide ring considered to be responsible for the antimalarial activity. So it is used in a treatment of malaria. We know that earlier we are using chloroquinone for a treatment of malaria. Later on, this parasite become resistant to that, and the people do have a lot of problems. And there are multiple drug resistance plasmodiums are also there. So we don't have a good treatment for malaria nowadays. What happened? Due to the artemisinin, we do have several derivatives of that, and they are most active against this Plasmodium falciparum, and we are getting rid of this malaria. So this artemisinin, it is very high efficient, very fast action, and this artemisinin is having very low toxicity and no side effects, no adverse reaction, and therefore it is termed as breakthrough in history of anti-malaria drug. If you remember this, in 2015, the first plant-based Nobel Prize was given to Tu Youyou in 2015 for her discovery of artemisinin from Artemisia annua. So this is also a very important drug, and it has been used in combinational therapy along for human cancers also. And it has been reported that I told you that terpenoid is very typical molecule, and it has been reported that terpenoid is this can be produced both in vivo and in differentiated in vitro cell lines. There is a report and the confirmation about this. And this artemisinin is present in the peltate gland of the plant Artemisia annua, and you can smell this artemisinin if you crush the leaves of this Artemisia into your hand, and you can just smell out and you can see the smell of this terpenoid. So this is a typical structure of artemisinin. That is a cis-terpene lactone, and this is a plant grown in a garden. What happened? This plant do have it is an aromatic herb, and it contains a cis-terpene lactone with the endoperoxide ring, and as it has a very low level of artemisinin, which limits its commercialization, and we need to have increase the content of artemisinin in this plant. And it has been found out from the literature that ionizing radiations are the most powerful technique for the plant improvement, especially in the crop plant. And later on, this artemisinin can be detected, quantified, and identified by various techniques such as TLC, Hplc, and GCMS. And this is a typical thing. What is the mechanism of artemisinin on a Plasmodium falciparum? The proposed mechanism of this artemisinin is it involves the cleavage of endoperoxide bridges by iron generating highly reactive free radicals, hypervalent iron oxospecies, epoxides, aldehydes, and decarbonyl compounds, which will damage the biological macromolecules, causing oxidative stress in the cells of the parasite. And this will damage the parasite membrane by covalently bonding to the membrane molecule. And this artemisinin will disturb the pathogen, and because of the stress and death of parasites can occur. So if you see from the initial point slides, I am telling you that the concentration is very low. So what is the need of plant derived artemisinin? Can we go for chemical synthesis of this? Yes, we can go for the chemical synthesis of this, but the cost is very low. Oh, sorry, cost is very high, expensive, and it is very complex. No, it has a cis-terpene ring, and there is a high global demand for this artemisinin, and we are not able to provide this. And there is a limited availability because of the low raw material, and we don't have the certified seeds. No. High level cell lines, which is, or the cell seeds, which will give, and high concentration of the artemisinin in the yield. I will tell you to the students of your interest, I will tell you that this plant is typical a Chinese plant, but this is grown in the Kashmir plateau up to the Chandigarh. Okay, and there another species is found common as a wild species in our forest areas also. So we need to go for high production cultures for this artemisinin. So it is very essential how to enhance the artemisinin concentration. It is very, very important. One of the method is to go by in vitro hairy root culture. Everybody knows it, by the gene transformation. And of course, go for cell suspension with the elicitation with some chemical elicitors as methyl jasconates and another one. And the most important is gamma ray. We know already what are the importance of the gamma, and we know what are the societal aspects of gamma radiations also. So it was a project. No, I'm dealing with the results and the facts, collaborating with this artemisinin. With my project, which I have done with the collaboration with the bar. So we went for the gamma radiations. And you know that action of gamma is very safe, and it is being, a lot of reports are there. And when you give a gamma radiation to any of the cell, what it will do, it will induce a free radicals, and these free radicals will act as a stress, abiotic stress, and it will triggers a stress response in the plant. And in this stress, there may be elicitations of the genes will be there. So mostly the gammas are used for this purpose, and we also use, there are a lot of references on different plants, not only on this Artemisia annua, but on different plants, what it will do with the different cell cultures and producing typical trapenoids or other typical type of the biomolecules or bioactive molecules with the help of upregulation of the oxidative stress. So in case of gamma, adaptability of the mutant genotype of Artemisia as a result of gamma irradiation in 3 locations with a different attitude has been reported in 2011. There are a lot of reports, no? Even in vitro artemisinin can be produced, artemisinin can be produced by Artemisia annua. You are, if you are able to trace out the things, you can see that terpenoids are the molecules which can handle the stress and even can come up to the hairy root culture or a fermentation or a bioreactor stage. It means that we have seen that the amount of terpenoid in a total biomolecule is about 75%, and it means that they are the best candidates to be drugged. So in our experiments, we have taken Artemisia annua variety Araucaria, and that is from CMAP by material transfer agreement. If you see, the seeds are grown on a plain MS medium. This is callus culture and shoot generations from the seeds. Okay, then this is a method to go how for root induction, root organogenesis. This is another good approach, that production of biomolecules, if they are low in a plant, you can induce some root. You transform the roots, and roots can be cultured on a bioreactor, and you can get a good amount of drug. In that, you have a different type of felicitors can be used here. If you use gamma, then you just grow them. This was a photograph I would like to show you, that the roots can be induced after the transformation, and that can be grown on a bioreactor. I'm going very fast because the time is very, very low. So in the first slide, we have seen that the callus and the shoot has been developed from the Artemisia annua. And what we have done it, we have given different doses of gamma radiation at the Bioatomic Research Center, and the dose level is from 5, 10, 15, 20, 25, 30, and 35 gyrus. And the culture to which we have given the dose is labeled as Z0, M0, and that is G0, M0 generation. And after every 39 days of interval, the subsequent subculturing was carried out, and they are labeled as M1 T1, M2 T2, M3 T3, M4 T4, and M5 T5. So about 5 generations, we have screened the analysis, the progress of trapenoid from stress to the final stage. We have screened out. And we, along with that, there's a lot of analysis techniques. We have seen what are the phenotypic variability of the callus, what is the survival rate of the callus, what is the lethal dose for the callus. After that, this callus is not able to survive. Well, we went for biomass measurement. We specifically focused on our point on artemisinin contained by HPLC, their antioxidant potential. Also, we went for total phenol and flavonoid because these are the markers of molecules, molecule markers, which can give you that when they will fight with oxidative stress, their concentration will increase. So this was first generation, I think is M0, yeah, it's M0 T0. See, after giving a gamma dose to the root and the shoot in the different concentration, the color of callus and the shoot goes somewhat blackened. They're in the shock, and this termed as M0 T0 generation. Now we waited for 37 days, and we went for observation of their callus type and the shoots. And along with the control, we compare the other type of doses of gamma, and we found that control was quite good as compared to the other plants which are in the stress. So this was M1 T1 generation. See, now you will feel something greenish color which is coming out. It means some stress has been released out, and they are starting to grow. Yes, and this is their characterization, growth characterization as compared to the control, and they are starting to develop from dark brown to pale brown, brown to light brown to pale yellow, yellow to somewhat like that. They started growing some compact, unfragile, less protrude callus to some normal callus they are trying to. So later on, we have gone for its fresh wet and dry wet. We come to know that those callus, those shoot cultures which are not able to sustain the the stress of gamma, they don't grow. So their weight is 0, 0. Okay. So we went for M2T2. You can see the change in the appearance of the callus. This is M3T3. I'm going very fast because the time is very less. This is M4T4. See the change in the appearance of the callus, and this is M5T5 most probably. Yes. Now, if these calluses are growing properly, and later on we are interested to see that in this up to 5 generations, now what is the fate of artemisinin or a terpenoid? And we firstly went to the analysis by the TLC, and for that TLC plates as a stationary phase and mobile phase with N-hexane is to acetone, 80:20 we have used it. Then we have extracted the leaf and the callus, and then we just ran it and we spread it with vanillin NLCI solution that is specifically for the terpenoids, and it shows pink colored band of artemisinin as compared to the standard. Now this is a TLC plates, and you can see in case of one is unstained, it is on A is under UV, and this is stained by the vanillin NLCI solution. So you can just check out the presence, and you can confirm the presence of the terpenoids or sesquiterpenes or artemisinin on the plate. Later on, what we have done it, we have went for its standard graph preparation by Hplc, and we use acetonitrile and Hplc grade 50:50, and the flow rate is 1 ml per minute, and the column temperature was set at 25 degrees. And later on, you can find that the artemisinin retention time is 7.3 minutes. And we prepared the standard graph of standard artemisinin, and the concentration of unknown samples can be calculated by the equation generated from the standard curve. This is a standard graph. Later on, we have done. This is a standard graph of different concentration of artemisinin from the standard concentration is 1 NT per ml, and you can see at 7.3 minutes, you can see the different peaks of artemisinin there. Later on, what we have done is, this is from a crude diagram. It's a pie diagram that is for concentration of artemisinin from callus in M0T0 to M5T5 generation. See, as compared to the control, you can see in M0T0 and control and up to M5T5 generations, and the dose, variable dose from 5 gyres to 35 gyres. You can differentially mark out the elucidations of terpenoids. Of course, they are not in a very significant and drastic straight one. They are different, no? Because we have used a callus, no, and callus are in an undifferentiated mass, and so this is a typical Hplc concentrations we have calculated from M0 to T0 to M5T5, and we can see that the terpenoids are producing best at the lower doses of gamma as compared to the higher gamma. So this is a standard along with all gamma irradiated callus and the Hplc in Hplc along with the standard artemisinin, and here we use Eclipse plus C18 reverse column, and about 20 ml or 20 microliters of the samples were injected, and you can see the different doses of gamma has a pronounced effect on artemisinin production, but the low doses can good elicitate the artemisinin. Yeah, this is another typical chart where after 2 months, 4 months, 6 months, this Artemisia is 8 months crop, and we have gone for 6-month analysis, and we found that in a 6 month, you have a good amount of artemisinin produced in a plant. So at the maturity, artemisinin is good. Later on, what we have done, this is a terpenoid. It's a smelling oil, and we went for its GCMS analysis, And the different type of doses, we extracted the oil from there, and we analyzed this GCMS, and we found that the different type of components are produced in a GC, reported in a GC. All of the component, the basic structure is a phenol. Basic structure they used to maintain is terpene. Most of the compounds which are reported shows the terpene structures. It means that terpenoids try to maintain its structure in a stress conditions also. See, camphor, it's a monoterpenoid, no? So all of this. Later on, as a stress, you always remember to relieve of the stress means increase of antioxidant activity, and then you have to estimate the total phenol as well as total flavonoid. I have only gone for a phenol because flavonoid, I'm not interested in presenting this to antioxidant one. If you estimate the total phenol by Foulencio calcio method, by using gallic acid as a standard 1 mg per ml, and at absorbance as taken at 765 nanometer. This is a standard graph of gallic acid in mg per ml, and it's a calibration graph. And you can see the total amount of phenol and compared to control as well as up to 35 grade. And you can see that the amount of phenol goes on increasing in shoot and root as compared to the control one. So this was a total table from M0 generations to M5 generations with all different type of gamma doses. You can find out that the phenol concentration goes on increasing in a lower doses, as we have reported that in a lower doses we got a good amount of artemisinin. So this is the conclusion that the terpenoid, this is a common that is artemisinin can be estimated using HPLC, and the mobile phase can be optimized best at 50:50 ratio, and the retention time of artemisinin is 7.3 minutes. And the low doses of gamma can elicitate the artemisinin. And the most important conclusion we can draw from this is artemisinin as a terpenoid is a very stable molecule, and it is able to sustain the doses of gamma, but still it can maintain its legacy from M0 to M5 generation. So at the end, I would like to thank Dr. Kamble as a director, and also Anupama Pathak, Professor Anupama Pathak Madam for these things. And I'm also wanted to acknowledge my Department of Biotechnology, Sandari of Amrata University. I'm also thankful to my collaborator, Dr. D.P. Fuljale. He's a retired ex-head Nuclear Agriculture Biotechnology Division, BARC, Trombay, and BRNSDA for the financial assistance. So I conclude my talk, and if anybody has a single question, you can just ask me. Thank you very much.
Thank you very much, ma'am. Now the topic is open for discussion. From audience, any question, please?
Thank you.
Thank you very much, ma'am. Thank you very much for sharing your profound knowledge with us. Your session was full of zeal and valuable information too, ma'am. Also, madam's session was informative. Wonderful talk, ma'am, for young researchers who are in initial stage of their research. And definitely it was very helpful, ma'am. Those who are having in their initial stage of research, how systematically protocol is designing and what skills we have to develop is very important in ma'am's talk. So also which tools or updated tools we have used and what are the traditional tools. So this is also important to see, to learn from madam's lecture. So once again, very thankful, ma'am. Thank you very much, ma'am. And definitely in future also, we would like to listen you, ma'am, in offline mode. Thank you very much, ma'am. Thank you. Now moving towards next session, that is for oral presentations. The offline paper presentation will be held in School of Life Sciences. So please join those who are giving offline paper presentation. They can go to School of Life Sciences, and for online paper presentation will be conducted over here. So please join for online paper presentation in a sequential manner. So now the moment has come to add a golden feather in the heart of success. We are going to invite selected young researchers for the oral presentation, and they will be getting 5 minutes each to showcase their research and discoveries, and 2 minutes for open discussion. Now I will call one by one. So be ready for oral presentations. We'll start soon for oral presentation. Before going to start, I will call name, ma'am, and later on we can. Okay. Now I will hand over Dr. Pathak, ma'am, for judges for oral presentation to introduce today's judges. Thank you, ma'am. Please.
Good evening. Today, I feel very happy to introduce two big scientists as chair and co-chair of presentation session, this online presentations session. Before I introduce these two formally, I am pleased to announce that both are alumni of School of Life Sciences. It is really a great feeling for me for having such a great alumni, and this will definitely inspire all students of School of Life Sciences. So first I wish to introduce Dr. Avinash Chaudekar, who is a 98 batch pass out. Dr. Avinash Chaudekar, currently he is working as a scientist of microbiology, Department of AIMS, that is All India Institute of Medical Sciences, New Delhi. Dr. Avinash works in section of virology. Dr. Avinash works closely with VRDL network COVID-19 response as a team leader during COVID-19 pandemic and viral diagnostic research. Prior to his appointment in 2021 here, Dr. Avinash served as Senior Research Officer in CDC USA funded research project SARS-CoV-2 transmission study and influenza surveillance as a part of elderly cohort study in rural villages, North India. Dr. Avinash earned a Bachelor of Science degree in microbiology from NSB College, Nanded, and Masters of Science from Biotechnology, that is School of Life Sciences, Swami Ramananda Tirth, Marathwada University, Nanded in 1998. Then Doctor of Philosophy in Microbiology from AIMS, that is All Indian Institute of Medical Sciences, New Delhi in 2012. He studied influenza and other respiratory viruses as a research fellow at Virology AIMS, New Delhi. So I feel really proud to have Dr. Avinash Chaudekar. Welcome, Dr. Chaudekar here. I welcome you. Now another person who is again big scientist, Dr. Mukund Adsul. He is presently working as a Senior Scientific Officer, DBT IOC Center of Advanced Bioenergy Research, Indian Oil R&D Sector, Faridabad, New Delhi. Dr. Mukund have already registered for, I think seven patents have been awarded in his name, US patents. He has published many papers in journals to those we used to call dream journals. So again, I feel very proud for having such a great alumni with us. Thank you, Dr. Avinash and Dr. Mukund, for giving us time. Students are very pleased to see you at screen. Now please act as chair and co-chair. So participants can present their presentation here, and these two eminent scientists will judge your presentations. Thank you.
Thank you, ma'am. Thank you, ma'am. Thank you, ma'am.
Thank you, ma'am. Thank you, Dr. Mukund sir, and thank you, Avinash Saudekar sir. Now, I would like to call one by one. Be ready. Amol Tungenwar. Next is B.D. Landge. Next, Sunita R. Mukkawar, Maithili Kasralikar, Patil Sheetal, Akib Nisar, Patil Sheetal, A.S. Dake, Nilkant Bhalerao, V.N. Vadekar, Suchita Bharambe, V.B. Fartade, Girse M.S., Vikas B. Zoshi, Aryas Moon, D.H. Dudhmal, Joita Kar, Patil Amol, Amita Godbole, and Shirsat SB. So, Amol Tungenwar is ready?
Yes, ma'am.
Yes. Now, you will be given 5 minutes for presentation and 2 minutes for the topic for discussion. So, you can start, please.
Okay, ma'am. Just wait. I will share my PPT.
Please share your screen. Amol Tungenwar.
Yes, ma'am. Is it visible, ma'am?
No, yet not.
I'm trying to. Just wait, please.
Next, be ready. B.D. Landge. What's the problem, Amol Tungenwar?
There is some internet issue. PPT is taking.
So, I will call next candidate. You can have arrangement of the nets, please.
Okay.
Next is B.D. Landge. Is ready? B.D. Landge, Yashwant Mahavidyalay, Nanded. Are you there? Next, Sunita R. Mukkawar, Department of Microbiology, B. Raghunath, ACS College, Parbhani, Maharashtra. Suchita Mukkawar is ready? Sunita Mukkawar. Sorry. Are you there, Sunita? Next, Maithili Kasralikar, Research Scholar, Department of Biotechnology, Gulbarga University, Karnataka.
Yes, madam, I am ready.
Yes.
Can I share my screen?
Yes, yes.
Am I audible, ma'am?
Yes, yes, yes. You're audible. You can start your presentation.
Okay. My PPT is visible, ma'am?
Yes, yes.
Yes, yes.
Yes. You can start.
So very pleasant good evening to all members. And first of all, I would like to thank all SRT conference members to give us opportunity for presenting our research in this, that too in the international conference on advances in bioactive molecules. So really we are pleased that our research is going to present on these dignitaries. Those are acting on international level. So thanks to all of you. you are giving us a chance. So basically, I am Maithili Kesradikar, PhD research scholar from Department of Biotechnology, Gulbarga University, Gulbarga, Karnataka, and I am working as a PhD research scholar under Dr. Ramesh Landankar, Professor, Department of Biotechnology. So my topic is phytochemical, in vitro anti-inflammatory, antioxidant, GCMS analysis of crude methanolic extract of Stachytarfeta mutabilis. So basically, healthcare and biotechnology is a very wide topic. So particularly considering healthcare and pharmaceutical biotechnology, my topic is coming under this category. So biotechnology has many divisions: agriculture biotechnology, animal biotechnology, pharmaceutical, industrial biotechnology. Vast topic is there. So regarding my topic, it is coming under pharmaceutical, particularly green biotechnology. My plant is the name of my plant is Stachytarfeta mutabilis. The plant is belonging to family Verbinaceae. Actually, this is a very rare family. This family is we can observe in the marshy areas, wet places. So flowers have a good nectar, and common names are velvet berry, porterweed, pink snakeweed. So in actually, if we see the observable characters of this plant, it is actually considered as a weed. Weed is generally unwanted plant, but we are considering, as per the literature survey, this weed has some very good bioactive molecules, and we have selected this plant so that we can isolate some bioactive molecule from this plant. And basically, as we all know, some of the drugs believed to be present in the plants are aspirin, artemisinin, colchicine, phytostigmine, vinblastine, vincristin. So in this line only we are working. A wide variety of chemical compounds are occurring naturally in plants, phytochemicals, and they have numerous medicinal activities. So based on this line only we are working. Pathway of plant derived drug analysis, already all the members are knowing. Plant extract, we have to select solvent system, whether petroleum, ether, chloroform, methanol, any choice of us. Then we have to isolate crude extract. After crude extract, again we have to go for antimicrobial, anti-inflammatory study, because based on our objective specific research. Then pure compound analysis, pure compound, after the pure compound analysis we have to go for isolation, GCMS analysis, NMR, FTIR, structural analysis, and after that pure compound, again we have to go for specific objective study, animal trials, cell line studies, gene expression analysis, flow cytometry. So that higher end techniques we have to go for check its any medicinal property. So this is the line of research of us. Then first of all, phytochemical analysis. So phytochemical screening is very important in identifying new source of therapeutically important compounds, so that we have to get the knowledge whether which compounds are present in our plant: alkaloids, flavonoids, phenolic compounds.
You have
Hello?
last 1 minute, please.
Saponins, steroids, tannins, terpenoids. So in our plant, methanolic extract has shown phenols, flavonoids, steroids, saponin, alkaloid, tannin, and glycoside, and lastly terpenoids. So we have selected methanolic solvent. So this is the screening of antimicrobial activity. 8 microorganisms we have selected. Among 8, 3 have not shown any type of activity. 5 microorganisms, clinically important organisms, they have shown the antimicrobial activity. As the concentration was increasing, zone of inhibition was also increasing. Next was we have done in vitro radical scavenging activity and anti-inflammatory studies. Three radical scavenging activity we have studied, in that ABTS, total antioxidant, and DPPH. So DPPH has given a very good results. Anti-inflammatory activity, as the concentration was increasing, percentage of inhibition by protein denaturation assay, it was increasing. Next was GCMS analysis. We have done TLC and GCMS analysis. 29 to 30 different compounds we got from this GCMS analysis. This is the structure, and these are the compounds of our that means through the GCMS analysis. This was last my MTT assay. MTT assay is for cytotoxicity assay. So we have done drug concentration versus cell viability and drug concentration versus cell death. So IC value also we have calculated. In IC value, it was 133. And these were the results for MTT assay regarding crude extract. So we are in trial of isolating the pure compound. Then these were the MTT assay results. We have done HeLa cells actually. We have done MTT assay against HeLa cells. So these were the HeLa cells. As my concentration, how it was increasing. HeLa cells were also showing the HeLa cells were getting decreased in the that means decrease in the amount of HeLa cells. So from all these research line, we can conclude that highest cell death was found at the drug concentration 805.2 microgram. IC50 value was 133.9. So in the present investigation clearly revealed that methanolic extract have the presence of variety of phytochemical, so that we can get anti-cancer compound. So we are still in a trial for isolating pure compound. Thank you so much.
Thank you, Maithili. You can ask questions, sir. Mukund sir and Avinash sir. Stop, Kela. Maithili, you can stop your screen share.
Yes, ma'am.
Mukund sir and Avinash sir, you can ask questions, sir. Please.
Maithili, can you hear me?
Yes, sir.
How many chemicals you have analyzed till date?
Sir, 29 chemicals we have analyzed, sir.
Antioxidant or anti-inflammatory?
Antioxidant property, sir. It is a combined. That means they are. We have actually checked with crude extract, sir.
Okay.
It is a combined activity.
Thank you. Next presenter.
Next candidate, Patil Sheetal, Assistant Professor in Department of Microbiology, Dayanand College, Latur. Maithili?
Hello. Yes, ma'am.
Your screen share, please.
Yes, ma'am.
Yes. Patil Sheetal. Are you present, ma'am? Patil Sheetal. Next, Akib Nisar.
Yes, ma'am, I'm here.
Yes, you can share your screen. You will strictly get 5 minutes only.
Sure.
You have to wind up your presentation in 5 minutes.
Yeah, just 1 minute, ma'am. Yeah, can you see the presentation, ma'am?
No, not yet, sir.
Is it visible?
Yes, yes, yes. You can start.
Okay. Okay, hello everyone. Myself Akib Nisar. Basically, I am going to present here my research entitled as Isolation of Prebiotics from wild vegetables in vitro characterization by antioxidant assays and human adenocarcinoma cell line. So basically, this research was done on Rajiv Gandhi Institute of IT and Biotechnology, Bharati Vidyapith. My corresponding author is Dr. Abhay Madhukar Hasulkar. So for introduction only, the prebiotics are the live strains of strictly selected microorganisms, which when administered adequate amounts confer a health benefit on the host. So we know about the probiotics. And likewise, they have many effects on different diseases, and they are known to improve the diseases, and they are very responsible whatever going on in your gut. And as well as prebiotics are the non-viable food component, and that actually support the growth of the probiotics. And their source of the prebiotics is fruits, vegetables, cereals, etc. So finally, symbiotics is also a term which combines probiotics and prebiotics, and which like combines probiotics and prebiotics, and it also helps to grow the probiotics in our gut. So if we focus the oxidative stress and role in disease generation, so oxidative stress may be responsible for many diseases like cancer and diabetes, Alzheimer's, etc. And the solution is that antioxidant mechanism of probiotics. So our probiotics actually helps to regulate these antioxidant mechanism, and they can scavenge free radicals, chelating metal ions are there that can be excreted from these probiotics. Regulation of enzymes and antioxidant metabolites are also there. So finally, we can say symbiotics are combined term probiotics and prebiotics, and basically they are known to have metabolic effects, changes in intestinal microbiota, immunomodulation, and many like pathways are there they can work on. Now, finally, as of now, many bulk investigations are there that actually have potential prebiotic that is oligosaccharides and that is pure. So here we are focusing on basically wild fruit and vegetables that may be a source of source of prebiotics basically as a oligosaccharide, and also there are some biotic compounds which in the form of phytochemicals and polyphenols that may help also to regulate any disease or fight any disease conditions. So finally, in this communication, basically in this research, we have selected 10 wild vegetable plants, and we have characterized isolated prebiotics characterized with the help of the lactobacillus strains. So in methodology, in brief, we have collected the basically and processed the plant materials. Total 10 plants were taken, and crude prebiotic isolation were done with hot water method extraction. And finally, we have isolated the prebiotics with the help of GI digestion assay with the help of pancreatin and pepsin enzymes. And we have also estimated the total reducing sugars. And finally, we have checked the growth of probiotics in presence of these isolated prebiotics. Simultaneously, we have checked the antioxidant activities of some bacterial strains, that is 4 lactobacillus strains. And we have checked the antioxidant activity about the cell-free supernatant as well as intracellular cell-free extract that is called ICFE. Finally, after screening this, we have formulated 20 symbiotic formulations with the help of some isolated prebiotics along with these. And also these symbiotic formulations were screened for the antioxidant assays, DPPH, scavenging ABTS, and FRAB. And finally, we have also maintained some of the H229 cell lines, and we have evaluated these formulations for their cell viability assay, cell adhesion assay, and we have also checked the microscopic observation of this lactobacillus adhesion on H229. Now, these are the list of plants we have selected. 10 plants are there. Okay, and this is the lactobacillus strain we have selected: Lactobacillus plantarum, 2 strains, and Lactobacillus bulgaricus as well as Lactobacillus lactis. All the plant sources were from the basically wild species of the vegetables. Now, this is the just for example of given Costospecious, like we how we have isolated the prebiotics. So these are Costospecious leaves. Now we have dried the leaves and grinded it, and hot water treatment was given to like isolate the crude prebiotic, and finally the purified prebiotic was found after the GI digestion assay. Now, results and discussion. So these are the results where we can see the highest prebiotic were isolated from the Umbilica officinalis, that is 3.25 gram. And the lowest was Solanum tarvum, that is 0.08 gram. Now, we have also performed the reducing sugar estimation for the prebiotics, and the lowest reducing sugar was found the Asparagus racemosus, and the highest was in Solanum nigrum. Along with that, we have also done the probiotic growth assay to check the prebiotic efficacy, if it is providing the growth of probiotics or not. And we have done with the control of with one control that is called inulin as a standard prebiotic. So we got the maximum growth in the Dioscorea elata, that is D. elata prebiotics in the presence of Lactobacillus plantarum. Now, again, we have performed the antioxidant activity of these lactobacillus strains only. So here you can see T3 assays are there: DPPH, ABTS, and ferric iron. In these 3 assays, in ABTS and ferric iron, you can see the supernatant is showing more antioxidant activities when compared to the ICFE. And when you see the DPPH, ICFE is showing the more activity when compared to the supernatant, with the one exception of LB7. Okay. So next one is the radical exchange. This is the antioxidant assay for the formulations. So here pattern was
your time is up.
Yeah, here a pattern was followed where the supernatant was showing the formulation was showing more antioxidant activity when compared to lactobacillus strains and prebiotic control only. This is called the synergistic effect we found in our study. The FRAP also showed the same kind of results. Again, we have performed cell viability, and where we got the more cell viability in presence of the prebiotics.
Please conclude your presentation. Please conclude.
Okay, fine. So this is the actually a microscopic observation we have received. Okay, addition of lactobacillus. Now, in conclusion, we can see the prebiotics are showing effects in the adhesion, cellularity, and antioxidant as well. So we can conclude that these wild vegetables can be a good source of the prebiotics as well. Thank you.
Avinash sir and Mukund sir.
Yeah, just 1 minute, ma'am. I just stop sharing my screen.
Yes, yes, sure, sure.
Akib, can you hear me?
Yeah, yes, sir.
On
Yes, sir,
what basis you have selected this plant?
Plants basically was selected on the basis of cultivability and availability on my local area. I am from Maharashtra, Pune, and also we have selected plants that is already known for their herbal activities in different diseases.
Okay, okay. But you already known.
Yeah, these are known actually plants for their herbal activities, but they are not being used for the prebiotics isolation as well.
Okay. Thank you.
Thank you, sir. Thank you, Akib Nisar.
Any more questions?
Next candidate is
Next
Patil
presenter.
Sheetal. Patil Sheetal.
Yes, ma'am. Yes, ma'am.
You
Yes,
can share
ma'am.
your screen.
Yes, ma'am. Yes, ma'am. Yes, ma'am. Is this my screen is visible, ma'am?
Yes, yes, your screen is visible. You can start. You will strictly get 5 minutes only.
Okay, okay, ma'am. Okay. My
Do you
topic.
have two presentations or one?
I have only one presentation, sir.
Okay.
My only one. Okay, my topic name is here I actually going to take antimicrobial activity of the Momodica chandadica. That is local name is the Carly, and also here I perform the it's nanoparticles. Okay. After that, here as we know that it is a green plant, green medicine hai, and a need for a green medicine, bitter gut. It is a name as a medicinal history of bitter gut, as you know that, and it is a medicinal. It has a medicinal application. This is the scientific classification of this bitter gut. That is a plantae. Order is the Cucurbita. Family is the Cucurbitaceae, and genus is the Momodica. After that, the aim of my practical aim of my this paper is the antimicrobial activity of bitter gut and production of silver nanoparticle. Object is to collect the bitter gut and extraction of bitter gut by using the different organic solvents. And also here we produces the silver nanoparticles, and here we perform the antimicrobial plate assay, and also here we determine the MIC. Then after that, for the here for antimicrobial activity, here we select the E. coli, then Pseudomonas aeruginosa, Salmonella, and the Staphylococcus aureus. Then method, here we firstly prepare the different organic solvents for the preparation of organic solvent. Here we select the different solvents, that is the methane, ethanol, like that. These solvents here we prepared. And then after that, here we prepare the silver nanoparticles, and also here we prepare the subculturing of the test organism, and also here we perform the antimicrobial activity of these selected pathogens, and MIC also here perform. These are the organic solvent of the different extract. For this organic solvent, here we use the ethanol, methanol, then petroleum ether, and then also ethylic and water. Also, these are the centrifuged organic extract.
Hello,
And
madam.
these are the photographic of the antimicrobial
प्रेझेंटेशन आता नका असेल ना मॅडम, ते नाव घेत आहेत ना एक एक झालं की 9व्या नंबरचा आहे.
Hello.
Yes, yes, yes. You can.
Can I mute them?
Okay, this is the photograph. These are the photographs of this antimicrobial activity of these pathogenic organisms, which is treated with the different type of the extract, solvent extract. These are the photographs of the E. coli, okay, which is treated with the water, ethanol, methanol, and also here we give the control. For this control, here we select the chloramphenicol. You can see here these are the zone of clearance. That is the zone of antimicrobial activity of these microorganisms against the these are the pseudomonas, which does not show the activity, which is also this is the plate of the salmonella. And in observation, this is the preparation of the nanoparticles. Here we prepare the nanoparticles of this Momordica. And in observation, you can see here, here we observe the maximum zone of inhibition. Here observe the extract of the nanoparticle. And for this extract, we observed the Staphylococcus aureus, which shows the maximum zone of inhibition against the Momordica, which is treated with the Momordica. And result of my presentation is the organic.
Can you zoom the? Can you
Yes,
zoom the
ma'am.
observations?
Yes, sir.
Can you zoom the observations?
For Staphylococcus aureus, silver nanoparticle shows the maximum zone of inhibition.
What is your conclusion from this table?
My conclusion is the all organic extract shows the antimicrobial activity, and order of this inhibition is like that. Aqueous shows the minimum inhibition. Then the methanol, ethyl acetate, petroleum ether, ethanol, and silver nanoparticle, which shows the maximum activity against the pathogenic organisms, sir. This is my conclusion. Means aqueous of this Momordica does not show the antimicrobial activity. It shows the antimicrobial activity, but as compared to the silver nanoparticle, it has the less activity. When we prepare the silver particles, it have the maximum activity, sir.
What
This
is
is
its
my
implications? What is the implications of this your study?
implication is that when we treat this Momordica with silver nanoparticle, it gives the maximum antimicrobial. It shows the maximum antibacterial activity against the pathogenic microorganism. Rather, this methanol, ethyl acetate, petroleum ether, ethanol, there is no need to treatment. There is no need to use of this organic solvent. We can directly when we use the silver nanoparticle. Okay.
Okay, okay, thank you.
Okay, thank you.
Next candidate is A.S. Dake, School of Life Sciences, SRTM University.
Yes, ma'am.
You will get 3 minutes, sir. You have to wind up your presentation and conclude in 3 minutes only.
Okay, ma'am. Yes.
Yes.
Can I share
You
the
can
screen?
share your screen, please.
Ma'am?
Yes, yes, yes.
Yes. You cannot start screen while your other participants sharing. Can you stop?
Sheetal, you can stop your screen sharing, please.
Yes, ma'am. I will stop.
Yes.
Yes.
A.S. Dake, please.
Yes, ma'am. Yes, ma'am.
Once
Is
your
it-
presentation is finished, please stop your screen sharing.
Yes. Is it visible, ma'am?
Yes, yes, it is visible.
Okay. Now my topic is antibacterial potential of marine Streptomyces strain RTMS3 against the Klebsiella pneumoniae. Introduction: the emergence of the multi-drug resistant Klebsiella pneumoniae, and this is caused by the Klebsiella pneumoniae. As we know, the Klebsiella has a multi-drug resistant pathogens. It is having the multiple diseases like hospital-borne diseases, urinary tract infections, and other hospital-borne diseases. The marine actinomyces has a novel antibiotic source, so it is not yet explored. So we need to explore the marine environment. The current study is the objective, current aim and objective is that to determine the antibacterial potential of marine actinomyces against the Klebsiella pneumoniae. The objective of this study is to isolate and screen the marine actinomyces for antibacterial activity, to identify the efficient actinomyces strain, to produce the metabolite from the selected strain, to purify and characterize the bioactive metabolite by using the techniques UV and FTIR techniques. This is the materials and methods. This is a picture showing the sample collections from the sampling area of the Ratnagiri of Maharashtra state. This we are isolating the.
Ajit, your slides are not moving.
Sorry?
We are just seeing your first slide.
Okay, sir. News. Now it is showing, sir.
Your slides are not moving. You just make it in a presentation mode, please.
Okay, sir.
Press F5. F5.
Okay, sir. 1 minute, sir. Now it's showing, sir.
Yeah, now it's
Yes, sir. Sorry, sir. This is the sample collection area. We are collecting the actinomyces from the Ratnagiri beach of the Maharashtra states. So we are isolating actinomyces. Total 9 isolates we isolating from the marine environments. Then procedure bacterial strain is collected from the nearest government hospital, Shankarao Chavan Medical Hospital, Nanded. And then we use it for the preparation for 0.5 microland standards. Then screening of actinomyces against the Klebsiella pneumoniae. And then identification of actinomyces based on the microscopic techniques. Then we are using the different media used for the production of the large scale production of the actinomyces strain which having the anti-metabolic properties. So after that we are purifying the extracted materials and used for the column chromatography and thin layer chromatography. The purification is done by the column chromatography using the different gradients. Then characterization of bioactive metabolites by using the UV and FTIR spectra. We are arranging the different range of the spectrums. The results we are got the screening of actinomyces against the Klebsiella pneumoniae. We are collecting the total 9 isolates from the marine isolates, which are showing the 4 isolates the zone of inhibition against the Klebsiella pneumoniae. Then the identification we are using the cover slip methods as we suggested by the cross 1989. So we got the gram-positive branching filamentous bacteria, which is spiral spore-forming chain. This is the Streptomyces species. So after that, we are using the different media compositions on the growth of the actinomyces. After the getting Artemia 3 strains, we are using 5 different media for the optimizations, and we got the yeast extract and malt extract agar, which are showing the highest percentage of inhibitions. So then the purification of antimicrobial compound by using the thin layer chromatography. Here it is showing the purified compound, which have been showing the single spot on the TLC plates, and which have showing second figure 6, which having the zone of inhibition. This characterization of the bioactive metabolite by using UV and FTIR spectra, the band which I have showing the compound having the polygenic activity against the Klebsiella pneumoniae. This is the graph showing by UV and FTIR. The summary of the study is the total 9 isolates of actinomyces was collected from the Ratnagiri sample, C sample, which having the tested for the Klebsiella pneumoniae. Among the 10 isolates, we got 4 strains which showing the potent activity against the Klebsiella pneumoniae. These isolates were optimized different media for yeast extract, malt extract media for which RTMI3, which showing the highest percentage of inhibition against the Klebsiella pneumoniae, which has identified as a streptomyces. Then the yeast extract, malt extract were used for the laboratory scale productions, purification of the bioactive metabolite by using the column chromatography and thin layer chromatography technique. The purification of these samples were done by the UV and FTIR technique. This is technique. Thank you.
Have you identified these isolates?
Yes, sir. We identified on the microscopic technique. This is the streptomyces species.
They are different?
Sorry, sir?
No, they may be one single or two.
Yes, sir. Single, sir. Streptomyces species. We are not identifying species level.
Okay.
Have you done about the microbiological characterization of these strains?
No, sir.
Microbiology.
Yeah, sir. We are not done by microbiology characteristics.
Okay, thank you.
Because they may not be a 9 isolate, maybe a 1 or 2.
Sorry, sir.
No, these are isolates, only streptomyces. No?
Yes, sir. The isolate is the only streptomyces.
Genus, yeah, species may be 1 or 2.
Yeah, species. We identify only one species. We are focusing for the large scale production, which showing the streptomyces species. The other species we are not identified.
Thank you.
Yes, sir. Thank you very much.
Please stop your
Next
screen.
one.
Yes, ma'am.
Next is Sunita R. Mukkavar. Ma'am, are you ready?
Yes, ma'am. Ma'am, am I audible?
Yes, you are audible.
Yes.
You will
Hello?
get only 3 minutes, ma'am. So
Okay.
please, you have to finish it within time span.
Okay, madam. Madam, is my screen visible?
Yeah, your screen is visible. You can start.
Okay, ma'am. So topic of my research, it is amino acid profiling of the bioprotein, that is single cell protein obtained from the saccharified banana agro waste. Just a minute, ma'am. It is not moving. Now here this yeast, that is Saccharomyces, we have taken for the production of the single cell protein, and which is grown in the agro waste containing medium, that is fortified with the saccharified banana agro waste. Now here the potential nutritional value of this yeast, it was evaluated again by means of the amino acid analysis and the protein levels. So basically the protein content, it was determined by the microgendal method, and the total amino acid content as well as the concentration of each amino acid in the single cell protein, it was determined by the LCMS and MS. So whatever the SCP which we obtained from this agro waste based media, it was basically found to be superior in terms of the lowered RNA content and the essential amino acids, suggesting that the agro waste, it can act as a very good substrate for the growth of these or for production of this single cell protein. Now I'll skip the introduction, only 2 lines I'll say. So because of the population growth, so major challenge it is just there for the food security, and because of which it is necessary to search for the new that is the avenues for the production of the nutritious food. And from that, now this single cell protein production, it is supposed to be the most promising technology for today's world. Now for this, again, basically a lot of hydrocarbon products or hydrocarbons, they have been utilized, or petroleum resources, they have been used as a substrate. But basically a lot of lignocellulosic materials, so including the agricultural waste, they are dumped in the field. And taking into consideration this matter, so in this present research, whatever the agro waste, that is the banana agro waste, a major heap of this agro waste is dumped in the Nanded district of Maharashtra, and that's why we thought that it should be utilized for the production of the value added product. Now objective of my study, that is to further reduction of environmental pollution, then production of the protein with the affordable price, good quality and very high nutritional value. The materials which are used, that is banana agro waste, that is the leftover leaves, pseudostem and the rhizome. It was collected from the banana plantation area. Then cell-free extract, that is
Please
cellulose.
conclude your presentation, ma'am. Please.
Okay, ma'am. Now the results of my presentation, see here, whatever the SCP which I have obtained. So these 4 media, the media 1, just a minimal media, which is a synthetic media. Second media, which is fortified with that of the saccharified leaves, and the minimal media without carbon and nitrogen. Media 3 with the saccharified pseudostem, and media 4 with that of the saccharified rhizome. So whatever the content of RNA which I have found in the product, this produced single cell protein. So basically, if you compare, here we will find that is the media number 2. So which is having the saccharified leaves as a substrate. It has shown a remarkable result, that is lowering in the RNA content. Then this slide, it is suggesting you that whatever these different the amino acid profiling which is carried out. So here promising results, they are related with the isoleucine, then methionine, as well as phenylalanine, tyrosine, valine, and lysine, where these 2 media, they have given remarkably high results as compared to the synthetic media. So indicating, so just I'll go for conclusion. So the SCP which is produced in the banana agro-waste based medium, basically it is superior in terms of essential amino acid content. Then it has lowered content of RNA, and apart from that, the saccharified banana leaves and the pseudostem, it has shown amazing increase in the amino acid content with respect to the isoleucine, phenylalanine, tyrosine, as well as the lysine, indicating that these agro waste, they can serve as a potential substrate for the high quality single cell protein production. So these are the results of my work. So thank you, ma'am.
Have you
Sunita madam,
analyzed the saccharified?
Yes, sir. Pardon, sir.
Yes,
Hello.
please.
Yes, sir.
Have you analyzed the saccharified?
Sir, after saccharification, we have checked the sugar content of that particular saccharified waste material. And when it is giving the, hello? Yes, sir.
What are these sugars?
Sir, it is consisting of the basically glucose, then rhamnose was there, and xylose sugar, it was in more concentration.
Whether this yeast utilizes xylose?
Yes, sir.
Okay.
Please stop your screen sharing.
Okay, ma'am.
Avinash, sir, you can ask questions, sir.
Mukhavar madam, it's a good presentation. What is a commercial application of your findings? Please, please
Sir,
explain.
Sir, if we just go for the pilot scale production, definitely I think the promising results we will get. Actually, this just on the laboratory scale, my research, it was carried out. And considering this huge amount of the agro waste, I think I can commercialize just if the feasible conditions are there. And definitely, I think at least for the fodder production, I can go for single cell protein production, and then in the next step, I'll just shift scale up for the human consumption.
Okay, good. Thank you.
Thank you, sir.
Please stop your screen sharing. Nilkant S. Bhale Rao. Are you there, sir? Nilkant S. Bhale Rao.
Yes, ma'am.
Yes, you can start your presentation. In 3 minutes, you have to wind up, please.
Okay, ma'am. Ma'am, is it visible for all of you?
Yes, yes. Right now, your screen is visible. You can start.
Okay, ma'am. Just. Okay. The today's topic for my paper presentation is the isolation and molecular identification of bioactive compounds. producing endophytic bacteria from the roots of Mangifera indica. It means the Alfonso. I have taken the Alfonso roots for the isolation of novel bioactive compounds. Myself is NS Bhalerao. Along with me, Professor DC Kamthane ma'am and Santosh V. Jadhav presented this paper. Okay. So all we know that the endophytes are the now currently we can see that the biggest source of the novel bioactive compounds which has something biotherapeutic values. Here I'm also the rationale behind this research work was to isolate screening and identifying the bioactive compounds producing endophytic bacteria. For this, I have used the materials like the roots and all the glasswares and the polymerase chain reaction machines. Here I have taken the test organisms, pathogenic organisms, Bacillus megaterium, Bacillus subtilis, Staphylococcus aureus, and E. coli. Here we have used many medias like the nutrient agar, tryptophan soya broth, nutrient broth, tryptophan soya agar, and for the sterilization process, surface sterilization process, we have used the sodium hypochlorite, ethanol, and for the extract process, we use the ethyl acetate. For the PCR purposes and the sequencing purposes, we use the many primers. Collection of the plant samples, Alfonso mango plant selected and the plant root material collected from the outskirt region of the Guhagar. As I am here, as I am from the Ratnagiri region, so I have choose this plant Alfonso from the Guhagar region, and the samples of the roots collected and transferred to the sample bottles, sterile sample bottles, to avoid any possible damages to the samplings. And then within 24 hours, we have processed the next steps. After the surface disinfection process by 1% sodium hypochlorite for 5 minutes and 70% ethanol for 60 seconds, we have transferred these root samples into the sterile tryptophan soya broth for the enrichment processes. After the growth of we got the growth heavy growth of the bacteria isolates, and by using colony picking methods, we actually seen for the antibacterial antagonistic properties against the pathogenic bacteria. We have found the one isolates which shows the highly antagonistic properties against the all 4 pathogenic microorganisms, and then we go for the strain identification of that particular potential isolates by using the 16S rRNA gene sequencing method and CTAP protocols. Then we produce the crude extracts from the same potential endophytes and go for the molecular identification. Here we came with the results. We have 8 isolate endophytic bacteria selected based on morphological biochemical characteristics and given the name MRB1, MRB2, MRB3, MRB4, 5, 6, 7, 8. Then determination of the potential isolates, antibacterial activity assay reveal the colonies from 4 endophytic bacteria can be the gram-positive and negative bacterial test organisms. Already as said that we use the Bacillus subtilis, Bacillus megaterium, Staphylococcus, and from gram-negative E. coli strains. MRB7 isolate inhibited all bacterial test organisms, and we used as a potential isolates.
Nilkant sir, please conclude your session, please.
Okay, ma'am. Yes. Just give me 2 minutes. Then we go for the morphological characterizations by microscopic and macroscopic observations. For this, we use the high media ready kit for this purpose. So this is the result from the primary screenings. Here we can see that the MRB7 shows some antagonistic activities. So we can decided to further go for the further studies of the MRB7 isolate. Here the colony morphologies and the gram staining figures. Then we go for the biochemical characterizations of that particular MRB7. Then we see the homology for the blast analysis and for the phylogenetic analysis purpose. Here is the 16S RNG sequencing we obtained from the resources. And this is the phylogram. Here we can see that the isolated potential isolates of endophytes is the Bacillus badius strain, which shows the 96% similarities. And the final is the conclusion. This study revealed endophytic bacteria, Bacillus badius strain B2S2, MRB7, which we have given the name, was isolated from the Mangifera indica L. alfonso. Ethyl acetate fractions metabolites maybe show high antibacterial activity against the Bacillus megaterium and all the pathogenic microorganisms. Furthermore, MRB7 produced bioactive and for future explorations, important to investigation and characterizations of all the compounds that may be used in the medical and pharmaceutical industries. Here, we are very grateful for the to the Research Center of Netaji Subhash Chandra Bose College, Nanded, Maharashtra, which is affiliated with Swami Raman Tirth Marathwada University, Nanded. Thank you, sir.
Okay, nice presentation.
How many sequences you sequenced, or how many isolates are sequenced?
Sir, we just got a best result from the one isolate MRB7, so we can do the we could do the for only one MRB7 isolate for gene sequencing.
Can you show that phylogram?
Yes, sir.
This phylogenetic tree is a rooted one or unrooted one?
Sorry, sir.
This phylogenetic tree is rooted one or unrooted one?
No, sir. I am not given the proper answer for this.
Okay. Thank you.
Okay. Thank you, sir. Thank you.
Please stop your screen sharing, sir.
Okay.
Vaishali Wadekar, ma'am. Ma'am, are you there?
Hello. Yes, I am there. Yes, I am there. Am I audible?
Yes, ma'am. You are audible.
Okay.
Please
Can I start sharing? Can I start sharing my screen?
Yes, ma'am. Yes.
Okay. Thank you. Is it visible?
Yes, ma'am. It's visible.
Yes, my screen is visible. Please tell me.
Yes, ma'am. Your screen is visible. You can start.
Okay. Good evening, all.
Continue.
You will get only 3 minutes, ma'am. Please,
Okay,
you have to wind up.
Okay, I will wind up.
Yes, sure.
Good evening, all. Myself, Mrs. Vaishali Wadekar. I am the faculty in the Government Institute of Science, Aurangabad. The theme of this conference is the advances in bioactive molecules. So, this is the right platform to give my some preliminary work, which has already done in my department. So the bioactive molecules, these are the molecules from biological origin. They are the parts, metabolites, or their byproducts, enzyme, pigments, etc. from the plants, animal, microorganism, algae, fungi, etc. That is some living organism. Just by narrowing our view on algae, now this algae is a major source of bioactive molecules. There are many references. Most of the bioactive molecules have been isolated and tested pharmacologically from the marine algae as well as from the freshwater algae. Here, a variety of marine algae also have been explored for their many compounds having antibacterial, antioxidant, anti-inflammatory, and radioprotective potential. Various algae and cyanobacteria have been screened for a variety of pharmacologically important bioactive compounds. These compounds are like the phenolics, flavonoid, tannins, phycocyanin. These are the pigments from that. Now, just I want to tell here that many African countries using the algae spirulina as a food and feed. They are using it as a nutraceutical, and because it is having the high content of the protein. So here, the scope of I am going to present my preliminary screening, in vitro screening of algae spirulina platensis from a very special natural aquarium, that is from the Lonar Lake. So the aim of my work is to explore the antibacterial, antioxidant, and anti-inflammatory potential of the pigment phycocyanin from the Spirulina platensis. So the objective of the work is isolation and characterization of Spirulina platensis from Lunar Lake. Then extraction of phycocyanin is the next step. Antibacterial, antioxidant, and anti-inflammatory activity exploration is the next part of my research. So the in introduction, I just want to introduce what is phycocyanin. Phycocyanin, it is produced from a variety of microalgae like spirulina. It is a photosynthetic in nature. Then it is blue colored, blue green colored algae pigments. Algal pigments, they are highly responsible for the health benefit. It has been already proved. So the spirulina species has been mainly used as a food and diet in various diet. These pigments are colored one. These phycocyanin are the small group of highly conserved chromoprotein that constitute the phycobilisome, a micromolecular protein complex. So this is the micromolecular protein complex which is visible on the screen, and it is mainly contain these phycobiliproteins are the alloproteins, APC, allophycoprotein, allophycocyanins, phycocyanins, and erythrophycocyanins. So totally it comes under the chromoprotein group, which is responsible for the photosynthetic activity of this Spirulina platensis. Now,
Madam, please conclude your slides, please.
Here is one side absorption and fluorescence has been shown. So the absorption of phycocyanin is between the 600 to 620, and it shows the fluorescence activity. Now, here the collection of sample is done during the rainy season from the Lunar Lake from in 2019 month of August. Then it is identified and cultivated. Then it shows, see here, this is the cultivation. The Spirulina platensis was successfully cultured in Jarcom medium for 20.
Madam, please conclude your slides, please.
Just 1 minute. So the morphological studies by morphological characterization, it has been identified by the Department of Botany. See, these are the microscopic structures. Just these are the microscopic structures, and this is the extraction by using UV light. So the maximum extraction is at the distance 10 centimeter from UV source, and the extraction was 1.37 milligram per ml. This is the highest extraction amount which I observed. This is the spectral analysis characterization. It is showing the 615 nanometer maximum absorption. So we confirm that this is the phycocyanin. Then this is the chromatographic analysis. Here is the A slide shows, A TLC slide shows the standard phycocyanin from the Sigma company, and the B is the our sample, and RF value is very much matching with that. So we confirm it. Then this antibacterial activity, I'm not going to explain everything, but see here the chart that is on E. coli, there is no any activity, antibacterial activity shown by the Spirulina platensis phycocyanin. So it can be used very safely as a food in many countries, and this is observed in case of the phycocyanin from our sample also. Then antioxidant activity is also showing with the, we compared it with the standard ascorbic acid, then standard phycocyanin from the standard company Sigma, and our phycocyanin in green color. So our sample is also combating well with the standard one. So
Madam, please conclude your session, because
last slide. This is the anti-inflammatory assay which is showing that the standard diclofenac sodium is taken as a standard one for the observation of the inhibition of protein denaturation. This is the assay used here. Our sample of phycocyanin is quietly equally inhibiting the protein denaturation. Hence, it is characterized as an anti-inflammatory also. And one more important thing I want to talk here is that this is showing the binding with the DNA molecule. So when the in future prospect is that when we use this phycocyanin instead of ethidium bromide, which we are using to visualize the DNA in the sample. So when if we can replace the ethidium bromide with this phycocyanin, as ethidium bromide is a carcinogenic and it is harmful to nature, and as this phycocyanin, it is having the fluorescent activity. If it is possible to in future, when we are trying for that, if this is having the proper binding with DNA molecule, then we can replace the ethidium bromide with the phycocyanin, and it will be a nature safely use of this molecule. So thank you for giving me the opportunity to present my work here. Thank you very much.
Please stop your screen sharing, ma'am.
Yes, just a minute.
Avinash sir and Mukund sir, you can ask question.
Yeah, no question from my side.
Okay.
Thank you. No question from my side also.
Then next is Suchita B. Bharambe. Are you present, ma'am? Suchita Bharambe. VB Fartade.
Madam, I am here. Hello, this is Suchita Bharambe.
Yes. You can start your presentation, ma'am.
Yes, just a minute.
Quickly 3 minutes, please.
Yeah. Yes, madam.
Yes.
So, good evening, all of the eminent persons
Your
present
screen
over
is
here.
not visible, ma'am.
I have shared actually.
Uh,
Yes, I will try it again.
Yes.
Is it
Now it's
visible now?
visible, ma'am. Yes, now it's visible, ma'am.
Okay. So I am Dr. Suchita Bharambe from Government Institute of Science, and today I am here to present a small topic from a research on phytase, that is purification and characterization of Aspergillus stapes FSP4 phytase. Actually, we know that phosphorus is one of the essential mineral for all the animals, as well as plants and microorganisms also, but here we are going to focus on animals, and it is available. The phosphorus is available to the plant to the animals from the plant-based diet, but it is not freely available. It is present in bound form in the plant in the form of phytate, and the monogastric animals, they doesn't have any endogenous phytase enzyme, so they're unable to digest it and make that phosphorus available to them, and it results into the loss of one of the important mineral resource, which may lead to the eutrophication problem. This is the structure of phytase, which have a number of anti-nutrient as well as significant nutrient factors, but because of lack of time, I just shift to the next one. So to avoid the anti-nutritive factor of the phytate and to get benefit of that organic phosphate available in the phytate, we have to the phytase enzyme is important. The phytase enzyme hydrolyzes the phytate molecule to inorganic monophosphates and the myoinositol phosphate, which make that phosphate, organic phosphate, available to the animals and fulfill their phosphorus requirement. So utilization of this phytase, it reduces the anti-nutritive effect of phytic acid, that is phytate, and it reduces the need of supplemental inorganic phosphate. Thus, we are saving the precious non-renewable inorganic phosphate resources. It also reduces the utilization of phytase in the enzyme in the feed supplementation.
Please conclude, ma'am.
Yes. Hello?
Please conclude.
Conclude.
Yes. Please conclude, ma'am.
So we just go for purification of the phytase, which is isolated from the Aspergillus stapes FSP4 strain. The crude enzyme is isolated, and it is purified by ammonium sulfate precipitation, acetone precipitation, and DAE cellulose method. So finally, the DAE cellulose column chromatography achieved the 48.03% purification, and it is then characterized. The characterization, it carried out by identification of molecular weight. The molecular weight by SDS gel electrophoresis, it is obtained approximately 43 kilodalton, whereas the native gel electrophoresis confirms the phytates present in that sample. The characterization is carried out on the on various basis, in that we just go for the optimum for optimization of temperature, and it is observed that at 50 degrees centigrade, the 50 degrees centigrade optimum, the phytase shows the optimum range. And thermals, when we study the thermal stability, it shows that the Aspergillus stachys have stability up to 60 minutes at 40 degrees centigrade, and it retain it at about 80%.
Conclude, please.
We also carried out the pH optimization.
Madam, please
the pH
conclude, conclusion, last slide.
As per whatever Aspergillus stachys strain FSP4 phytase shows, it is characterized. It shows good indication for utilization of phytase in broiler feed, which is our final application. The approach for this production and application of phytase can be environment friendly and cost effective. The decrease in production of cost is obtained because of the use of cheap agro residues. In addition, the utilization of agro waste will be helped to alleviate the smoke pollution problems. So basically, we produce this phytase enzyme for its application in poultry feed, and we get very good result regarding this. That's it. Thank you for providing me the opportunity to present.
Next. Please ask questions, sir. Avinash sir and Mukund sir.
Yes. Apart from making phosphate-free, is there any applications of these phytase? Other applications?
Phytase have applications. Actually, in the making available that phytase phosphorus.
Because minoxidil is a immunological molecules.
It has yes. Your voice is not clear, sir. Yes.
Okay, apart from making phosphate free,
yes,
whether this phytase has any other applications?
Sir, as it is a hydrolyzing enzyme, it has this application in all the monogastric animals.
Okay.
Please stop your screen. Next, VB Fartade.
Yes, ma'am, I'm present.
Are you there, sir?
Yes, ma'am, I'm there.
Yes. You can start your screen sharing. Strictly 3 minutes, please.
Okay.
You can conclude in 3 minutes, please, sir.
So, good evening, everyone. I am Fartade Vilas, a PhD student working in CSR, National Institute of Oceanography, Goa. Today, I am going to present my work, which is done on corals, mucus property, antioxidant properties, and quorum quenching properties present in the mucus. So, corals are the marine lower invertebrates which are symbiotically associated with the zooxanthellae, which secretes the mucus, which has the defense properties against the variety of coral pathogens. So these are the my study. These are the major coral reef regions of India. Among that, my study region is this Gulf of Mannar, from where I have collected samples and done this study. So this is the mechanism of how this enzyme enzyme cascade activated in the invertebrates. So this peptidoglycan and this kind of signaling molecules activate the serine protease cascade, and this activates the phenol oxidase enzyme, which is a very important enzyme which activates the melanin synthesis pathway, which activates the others all antioxidant enzymes like SOD catalase peroxidase. So during this my study, I have taken these 4 mucus samples, which healthy acropora mucus, diseased acropora mucus, healthy porous and diseased porous mucus. I have compared this on native pest gel. I have checked this SOD superoxide dismutase activity on native phase gel. So I have found all this in all these samples SOD enzyme, which is very effective antioxidants against the superoxides. Also, I have checked the catalase enzyme activity present in all these four mucus samples, which is the another important enzyme which shows the antioxidant activity, which degrades the hydrogen peroxide which is present in the mucus. So again, I have characterized this in hydrogen peroxide activity of mucus with this 3% hydrogen peroxide from all mucus. I got the activity. So along with this superoxide dismutase and catalase enzyme, I have also checked the other antioxidant enzymes such as phenol oxidase and peroxidase enzymes, which are present in the all these mucus samples, which are highly expressed in the particularly in disease. So I have checked this antioxidant, non-enzymatic antioxidants, disease and coral, healthy and diseased coral mucus. So this is the reducing power of this coral mucus against the reactive oxygen species, which can add the hydrogen or hydrogen molecules or remove the addition of or removal of the hydrogen and electron molecules to reduce the antioxidant trace in the mucus. So along with this enzymatic antioxidants, I have checked non-enzymatic antioxidants such as H2O2 scavenging activity, hydroxy scavenging activity, DPPS scavenging activity, and chelating capacity of the mucus, which showed more in specifically more in the tissues compared to mucus, and more in disease samples compared to healthy samples. So this is the AHL quorum sensing molecule present in the mucus. In this figure C, we can see the healthy acropora mucus, which showed the zone of clearance against this quorum sensing molecule present in the sample. So also I have checked the lactonase enzyme activity of the mucus, which all four mucus which I have studied showed the lactonase enzyme activity against the quorum quenching molecule. So also I have checked the coalescing pigment production in the presence of mucus inhibitors. Here also we can see this healthy porous mucus and healthy acropora mucus, which showed the quenching property against this quorum sensing molecules. So also I have checked.
Please conclude, sir.
Also, I have checked the actinomycete, which is the coral beneficial bacteria, which showed the activity against the coral pathogens. And also, I have checked the mucus activity against the Staphylococcus aureus, which also showed the effective antimicrobial activity against the Staphylococcus aureus. So during in this my study, I can conclude this coral mucus has showed this effective antioxidant properties such as H2O2 scavenging, OH scavenging, DPPS scavenging, and others enzymatic antioxidants, and also it shows the quorum quenching property, which is very important in case of against coral pathogens. These are the references. Thank you. Thank you.
Please stop your screen sharing. Avinash sir and Mukund sir, please ask questions, sir.
No more question from my side, please.
Avinash sir.
No question from my side. It's a good.
Okay, thank you, sir. Girse MS. Next. Girse MS. Are you there, sir? Girase MS. Next is Vikas B. Zoshi.
Yes,
are you there, sir?
Yes, ma'am.
Yes. You can start your screen.
Okay, ma'am.
You will get only 3 minutes.
Yes, whether my screen is visible?
Yes, sir. Yes, sir. Your screen is visible.
Okay. Good evening, dignitaries. I, Vikas Joshi, research scholar from School of Life Sciences, Swami Ramanand Tirth Marathwada University, Nanded. My today's topic is extraction of biodegradable polymer of halophilic bacteria that are isolated from mangrove sediment. This is the abstract which we had submitted already. The introduction of my topic is, as we know that the mangrove, these are the halophytes, and they are having ability to tolerate high salinity, and they are also flourish many kinds of bacteria, specifically those bacteria who are having ability to grow in high salt condition. the present investigation or present.
Vikas, sir, you can directly
yes.
go to material methods and result discussion,
Yeah,
sir.
ma'am. Yeah, ma'am. The present investigation sampling site is named as Bordi, which is a green belt tribal zone in district Palghar. This is the sampling site. This is the mangrove region, and here me and my guide who collected the sample, materials and method. All the sediment sample, the composite sample, we are enriched, and we isolated the halophilic bacteria by inoculating the enriched sample into the medium containing 5, 10, 15, and 20% of salt. And we the isolate which we are getting during this research work, that isolate we are checked for the PHB producing abilities. The screening for PHB producing abilities, the isolate were checked for PHB granule into their cytoplasm by using the lipophilic stain Sudan black B. After that, we are inoculated the isolates into the Davis medium, which with carbon source as a dextrose, and we kept it for 3 days at room temperature on a rotary shaker, 150 RPM revolution per minute. After 3 days, we are extracted the precipitate of PHB, not exactly PHB right now, but the extract we are getting from the centrifugation. After the centrifugation, that extract we are proceed for the different test. The first test which we are performed, that is sulfuric acid test, where 2 ml of sulfuric acid we are added into the extract and heated for 20 minutes at 100 degrees Celsius in a water bath to check the color change. Also, the extract was used for the thin layer chromatographic technique. Also, we are checked the melting point of the extract, and we are sent to the Center of Excellence WAPI for FTIR analysis. A result out of 7 selected isolates, only 3 isolate, that is BMS2, BMS4, and BMS5, were appeared with lipid inclusion granules. The inclusion granules were stained blue-black, whereas bacterial cytoplasm was stained pink. Colonies of these 3 isolated BMS2, BMS4, and BMS5 were retained the black color of Sudan black B, stain on 5% nutrient agar plate. These are the some pictures. This is the first picture which shown the granules present in the cell. These are the 2 pictures of plates that showing the granules present, granules produced by the colonies. This is the precipitate which we got after the centrifugation, and this is the H2SO4 test. Initially, we appear this tube like this, and after addition of H2SO4, this is the final result or conclusion, where the TLC plate was sprayed with the iodine vapor. A greenish black spot was appeared, and the RF value was approximately the 0.70. The RF value which indicates the white powder is of PHB, and the melting point is.
Please conclude your session.
Yeah. Conclusion, ma'am. Yes. Conclusion is going on.
Yes.
And the melting point of that melting temperature of our extract was found to be the 172 degrees Celsius. This is the FTIR analysis, and according to the standard data, that IR analysis, the peak which is shown by the graph, that clearly indicates that our extract was containing the PHB peak at 1262 and 1725 per centimeter. That shown the PHB was present in our extract, but there is some impurities in the our extract. It needs to be again purified. Acknowledgement, and I acknowledge my deep gratitude to the Honorable Vice Chancellor of Swami Ramanand Marathoda University, also the Principal of Gokhale Education Society, N.B. Mehta Science College Bordi, Director School of Life Sciences, Swami Ramanand Marathoda University, my research guide, Dr. Mrs. Anupama Patak Madam, and organizing committee. Thank you very much.
You can ask questions, sir. Avinash sir and Mukund sir, please.
How much maximum?
Sir.
Yeah. Yes, ma'am. Hello.
Have you isolated this PHB?
Yes, sir.
Have you isolated the PHB?
PHB?
How much the maximum production you get?
Sir, this is just a small scale level. This is one of the part of my PhD research. Production and application part is left here. I'll try my.
Okay, next one.
Avinash sir.
No question on my side.
Thank you, madam. Amrita madam.
Aryas Moon. Next candidate is Aryas Moon. Joita Kar. Joita.
Yeah, I'm here, ma'am. Yes, ma'am, I'm here. Can you hear me?
Yes, ma'am.
Yeah,
You're
okay.
audible. Yes.
Okay.
You can start your screen sharing, ma'am.
Okay. Okay, can you see my screen?
Yes. Your slides are visible, ma'am. You can start.
Okay, then. Yeah, this is Joita Kar from the Department of Biotechnology, Mizoram University, Pachunga University College Campus, Aizawl, Mizoram. And I'll be presenting on the topic cyanobacteria derived metabolites for bioactivity potential and acute respiratory tract infections. So first of all, I will start off with cyanobacteria metabolites for acute respiratory tract infection. So as we all know, we are currently suffering from this global pandemic that is COVID-19, and it is the preeminent global pandemic among infectious disease in the 21st century. So this created a global health crisis that had a deep impact on the way we perceive our world and everyday lives. And it had also a significant effect on sociological, physiological, and economical effect also. So there is as we all know, there is a direct correlation between COVID-19 and acute respiratory tract infection and also acute respiratory distress syndrome. And severe cases of COVID-19 infection leads to acute respiratory distress syndrome and pneumonia, which can prove fatal to infected individual if they are not given treatment in proper time and if they are immunocompromised. Also, due to viral infection, there cases of secondary bacterial infection also arises. And if they are not treated properly in time, it can also be fatal to the infected individuals. So this is not the first time that we are suffering from global pandemic. But this is the largest scale, obviously the present one, but previously SARS-CoV was identified in 2003 also in China, and after that in Middle East, MERS-CoV was also identified in 2012. Apart from that, influenza virus H1N1 was also identified back in 2009 at Japan. So this is the data released by WHO. Where you can see that clinical pneumonia incidence in children younger than 5 years has been rampant in our country. Rajasthan, Gujarat, Madhya Pradesh, Uttar Pradesh, Bihar, and Jharkhand are the countries that are suffering the most in this sense. And northeastern countries and other parts of the countries have lesser cases, but still it's quite alarming that this many children are suffering from clinical pneumonia at this rate. So could the most primitive organism solve this problem? This is the question that arises. So cyanobacteria are one of the most primitive photodiazotropic and ubiquitous in distribution organisms. Various species are already known to produce various metabolites of well-established bioactivity. So obviously we already know that they are, cyanobacteria produces bioactive molecules that have anti-cancer, antiviral, antifungal, antimicrobial activity that we already know. So the problem actually with this is that the bioactive molecules, they are generally cytotoxic to cell lines. So we should aim to find bioactive molecules that doesn't target biological process and do not occur in animals, especially mammals. So the hypothesis is, can cyanobacteria antimetabolites be exploited?
You can
for
directly go to material method and result, ma'am,
yeah,
please.
this is all the conclusion now. So yeah, we isolated nostoc species from the Kamble Lake of Mizoram, and then we use those extract to see the antimicrobial activity and then algicidal activity and antifungal activity. So most of the time, methanol extracts were the most effective one. And after that, dichloromethane and isopropanol extracts were the most effective ones against the algicidal activity that showed algicidal activity. And regarding the gram-positive bacteria, Bacillus subtilis and Bacillus pumilus, DCM, that is dichloromethane and isopropanol, had better activity than other ones. The same was seen in Bacillus pumilus also. And this is the two fungal organisms where the extracts were used, methanolic extract, ethanol and water extract, and dichloromethane and isopropanol extracts were used against them, and they showed quite good activity.
Please conclude, ma'am. Please conclude.
This is the conclusion. So we have identified nostoc from Tamdile. So preliminary findings suggest that the extract isolated from cyanobacteria showed antimicrobial activity against various organisms to date, and we will try to find out more through other experimentation, and then we will do cell line experimentation and mice experimentation also in future. So this is the acknowledgement. I would like to extend my acknowledgement to Professor S.K. Mehta, that the professor from Department of Botany, Mizoram University, and my co-PI and PI, Dr. Prashant Singh and Dr. Mukesh Kumar, and my two seniors, Ruthi Lalmanswili and Senjang Momi from the Department of Botany, who helped me out with these experiments, as this is a new field which I'm working on, where I haven't previously worked on. That is all. Thank you.
Yes. You can ask questions, sir. Mukund sir and Avinash sir, please.
Okay. No question from my side, please.
It's a good presentation. I want to ask only one basic question. What is the bioavailability of the metabolites and what is the bioactivity? Okay, can you make a difference? Tell me the difference. What is the bioavailability of the metabolites in terms of you're saying it will be used for the pneumonia and all? So what is the bioavailability? Can you define? Give me the.
Okay, sure, sir. But, last night I was reading a research paper. In there, I saw that bioavailability experimentation they did on mice actually. So mice, they extracted the blood, I think, from mice, and then they checked the bioavailability of that compound in the serum. So
no, actually, I'm not asking in like practical experience. Just I wanted to ask you, what is the bioavailability of the metabolites? If you are giving any drug or any plant extract or any drug, any Ayurvedic drug or something, what is the bioavailability? Just tell me the definition.
Okay, yeah, that definition I'm not sure about, sir. I'm so sorry because I don't want to give a wrong answer.
What is the bioactivity of the metabolite?
Means its activity against a particular organism, how much it's like, if it's increasing the growth of that organism or if it's inhibiting the growth of that organism, that will be the bioactivity potential, I think. Because in this research which we did, the water extract and was in some cases increasing the concentration of that organism while working on it. So bioactivity will be that either it will increase the concentration of that organism or activity of that organism, or it will inhibit the activity of that organism, a particular metabolite or anything.
In your hypothesis, you are written ARIs. What is ARIs?
This is acute respiratory tract infection, sir.
Okay. Thank you.
Okay, thank you so much.
Please stop your screen sharing, ma'am. Patil Amol.
Hello, ma'am. I'm Dr. Girase. Am I audible? Can I give my presentation, ma'am?
Okay. You can start. You have to wind up in 3 minutes, ma'am, please.
Yeah, sure.
Yes. You can start your presentation, please.
Also, I am waiting, ma'am. Sharad Gangavne.
Good evening. I'm Dr. Girase. Today, my topic of presentation is active chitin recovered from shrimp cell by using microbes. Origin of the research problem. We know that chitin is the second abundant biopolymer. It is present in nature. Every year, around 10⁻11 tons is synthesized in skeleton and fungal cell walls, spines of diatom, shells of crustaceans, shrimps, crabs, lobster. In shrimp, this is one of the waste product, and this can be utilized for recovery of chitin, which is a natural polymer. Because this chitin and its polymer or its derivative chitosan. They are having application is many field like water treatment, agriculture, textile industry, biotechnology, food and health industry, cosmetic and biomedical applications are there. So we can use this material from this source. Now current chemical methods for this recovery are from shrimp cell, demineralization with HCl, deproteination with NaOH, and then chitin is recovered. Now drawback of this method is that all these chemicals are strong chemicals like HCl and NaOH, and many times this chitin undergo denaturation. And whatever protein is recovered from this waste, that is mixed with the chemical like NaOH, therefore it cannot be used as a feed. So that is the drawback of this method. So in my work, I have selected the microbial deproteination of shrimp cell. Means instead of NaOH, I have used protease producing microorganism. So shrimp cells were cleaned, washed, dried, and they disintegrated into size of 8 to 10 millimeter. Then organism was isolated from loner lake and it was identified as Vibrio magnicovi. And by using this organism, 34.95% protein was recovered, and that was not contaminated with any chemical. And this recovery is around 94.46 recovery from the shrimp cell. Now demineralization can be carried out by using HCl. That was another experiment. Instead of HCl, I have used lactic acid producing bacteria for the recovery of chitin. So from the shrimps, eco-friendly method of chitin recovery has been developed. Now when I have studied the structure of this chitin, which is recovered by microbial method and which is commercially available, then it is observed that they are quite similar. And this data is obtained from the FTIR analysis. When FTIR analysis is checked for biologically recovered chitin and chemically recovered chitin, then they are having similar bands, and it proves that the chitin which is recovered biologically, that is of same quality as the chemically recovered chitin. Even X-ray diffraction studies have confirmed that they are having similar peaks. Both the chitin shows 2 theta value at 21.73. They reported the maximum intensity at 2 theta 21.1 and smaller peaks at 29 observed in biological recovered chitin, and it indicates that some impurities are there with biological recovery. So we can further work on this recovery process. So this is about the X-ray diffraction studies. From this crystallite size has been obtained. Now conclusion of my study is 30.09% chitin was obtained from shrimp cell using biological process. Crystallinity index of this biological chitin was 51.10. Biologically recovered chitin has crystallite size of 32.2 nanometer. And from the similarities of FTIR bands and the peaks which are obtained in XRD, it is concluded that biologically recovered chitin is very similar to the chemically recovered chitin. So these are the references which are used by me. Thank you for patient listening.
Please stop your screen sharing, ma'am. You can ask questions, sir. Avinash sir and
How much
Mukund
total protein you recovered?
The total protein recovered was around 94.46%, and in quantity wise it was 34.95 gram protein percent.
Nice presentation. No questions.
Thank you. Thank you, sir. Thank you.
Thank you, ma'am.
Please stop your screen sharing, ma'am. Patil
Yes.
Amol. Are you there, sir? Patil Amol.
Hello, ma'am.
Yes.
Amol Tungewar, in place of my name is Sharad Gangavne.
Okay.
I am waiting from.
Okay. You can start your presentation, sir.
Yes, ma'am. Yes. Thank you.
3 minutes, sir. Direct, you can go to material methods and conclusion, please. You can start your screen sharing, sir. Sharad, sir. Sharad, sir. Sir, तुम्ही शरद You can start your screen sharing, sir. Sharad. Unmute yourself, sir. Sharad, sir. Please unmute. Next, Patil Amol. Are you there, sir? Patil Amol. Amita Godbole.
Yes, ma'am.
Yes, you can start your presentation, ma'am.
Yes, ma'am.
You can start your screen sharing.
Yes, ma'am.
Madam.
Direct, you can go to material methods and result discussion, ma'am, please.
Yes, ma'am.
Madam Amol Patil, sir.
You please kindly wait.
Okay, madam. Okay.
Ma'am, is my screen visible?
Yes, your screen is visible.
Yes.
Please start your presentation, ma'am.
So, I am Amita Godbole from Nagpur University. Today I will be presenting a small initial portion of my PhD work which I carried out. The title of my work is Isolation and Diversity of Marine Actinomycetes from West Coast of Maharashtra, Screening for Biological Activity. So plan of work is first sample collection, different pre-treatments, then selective isolation of actinomycetes to obtain pure cultures of actinomycetes and screening of them for bioactivity. So all the culture media and drugs we procured from High Media and test organisms I procured from LIT campus and Hislop College, Nagpur. These are the samples. Sample 1 is mangrove forest near Gorai Khadi, Mumbai, Maharashtra. Sample 2 is Malgund seashore, Konkan, Ratnagiri district, Maharashtra. And sample 3 is Guhagar seashore, again Ratnagiri district, Konkan, Maharashtra. So all the samples were collected in the month of June, and then they was collected from the seashore at the time of low tide. So the seabed was exposed, and mangrove sample was collected from the roots of mangrove tree in the mangrove forest. So let us come to the sample 1, that is the mangrove forest. This is the site from where I collected the sample, Gorai Khadi. Characteristics were it was dark brown, sticky, muddy, pH was slightly alkaline 8.5, and it has had a seawater like fishy smell. Then sample 2 is this is a Malgund beach. The peculiar characteristic of this beach is because in the monsoon season, the entire seabed gets covered with a black sand and it remains like that for till September. So we selected this period, as well as the Malgund beach and the Guhagar beach and mangrove region are not still explored for the isolation of bioactive metabolite producing actinomycetes. So that was the reason behind selecting the season and the site for these samples. This is sample collection. Characteristic, it was a black sand. It was very, very heavy, very fine and shiny particles. It was very soft and free flowing heavy sample of pH 8, the Malgund one. Then this is the third sample, Guhagar beach, which is located just next to the Malgund beach, but the sample first sample is a Malgund beach, which is quite dark, heavy and shiny, smooth particles. These are the different pre-treatment concentration 100 microgram per ml and nystatin 50 microgram per ml. All the isolation media were initially prepared for isolation by
In between, your voice is interrupted, ma'am. You're not audible. Amita Godbole. You're not audible, ma'am.
forest. These are the pure cultures. This is the isolation of screening plates for Malgund beach. These are the pure cultures of Malgund beach.
Good, ma'am. Please continue.
Yes, ma'am. Yes. This is the Guhagar beach. The pure cultures of Guhagar beach I obtained on screening plates where they failed to grow up on subculturing. Hence, no pure culture was obtained from the Guhagar sample. Then they were screened for antimicrobial activity by a couplet method. These were the series of colors they produced: the golden yellow, the dark brown, and the pink. They are separated and the ethyl acetate extract of cells as well as the broth, as we can see here, was tested. It was concentrated.
Please conclude.
Yes, ma'am. The result is total number of isolate were 38. Best suitable treatment is 55 degree for 1 hour, and for Malgund and other marine samples was air drying. Maximum numbers were obtained 20 from mangrove forest, followed by Malgund, and last one is Guhagar. Starch Casey Nagar gave me the maximum isolates. These are the results. Bioactivity was very poor. I got many isolates, but many of them were lost during the COVID period. The 11 cultures we could save that were tested for antimicrobial activity, but activity was very poor. So M11 produced yellow color, which is a promising isolate, and some pigment also produced by different isolates. Some rare genera were also knocked. The results of identification, we sent the samples which are promising to the Ncmr laboratory Pune and identified them by 16S rRNA technique, and they are identified as Streptomyces freddi, Nocardiopsis. This is the again species which is knocked out, which is a rare species, and another two are Streptomyces species. So to conclude my study, actinomycetes which are obtained from Indian oceans and mangrove could be a promising source of bioactive metabolite. It was really difficult and challenging to obtain and cultivate these marine actinomycetes in normal laboratory conditions. We need to have different techniques to knock rare actinomycetes from marine sources, and need to understand the chemical and physical changes that take place in the sea to design isolation media. Further study of selected isolates could lead to the promising results. These are the references. Thank you so much, sir.
Can I ask questions, sir? And
Okay, it is just a preliminary data. No questions, please.
Thank you, sir.
Hello, ma'am.
No question from my side. Thank you.
Okay.
Thank you,
Please
sir.
stop your screen, ma'am. Please
Yes,
stop
ma'am.
your screen.
Yes, ma'am.
Next is Shirsat Shivaji Balaji. Shirsat SB. Next candidate is Dr. Nausheen Sadaf. Are you there, ma'am? Nausheen Sadaf. Sir, hai ka?
Hello? Hello?
Hello?
Can you hear me?
Yes, yes, ma'am. You are there.
Okay. Shall I start sharing the screen?
You can start your screen sharing, ma'am. You will get only 2 minutes, ma'am, please. Due to time constraints, we are already getting late.
Yeah, sure.
Yes. You can directly go to result discussion and material methods, ma'am.
Yeah.
Yes, yes, ma'am.
So, very good evening to all of you. This is Dr. Nausheen, and my topic is PHB production from dairy industry soil isolates using whey as carbon source. So this is my abstract where I have attempted to produce PHB granules from the bacterium which were isolated from soil that was collected from dairy industry. I have collected sample from the dairy industry MIDC area Nanded. The isolation was done on the normal nutrient agar medium, and the organisms were identified by Burgess manual. The staining performed by using Sudan black pea and Nile blue A staining. The production was done by using the pure sugar as well as 2% whey as a source of crude sample, crude carbon sample. So the extraction was done by using a method which was described by Abdul Karim and Agior et al. And in case of molecular characterization, I have done different techniques like FTIR, GCMS, H1NMR, C13NMR, LCMS, DSC, and Hplc. This is a graph that shows the maximum absorbance of PHB at 235 nanometer. These are the observation table that shows the isolate T1 shows the maximum production of PHB. The optimum temperature for production, it was around 35 degrees Celsius. The pH was 7 and duration was 48 hours. And these tables clearly indicate that the PHB production is more in case of crude carbon source, that is whey, when it is compared with the pure sugar, that is lactose. So these are the most prominent state, that was the T1. It was identified 60 RRL sequencing. The biodegradation is also done by using in vitro and in vivo studies, and the organism, the produced polymer was clearly degraded both in vitro and in vivo condition. So here I conclude that we can isolate microorganism from dairy industry soil, and that microorganism can produce PHB by using whey as a carbon source, as a sole carbon source. The produced PHB can be biodegraded in both in vivo and in vitro condition. That's all from my side.
You reported PHB in percentage.
Yes, sir.
Right?
54%.
Okay. Comparison to what? What is
Comparison
100%?
with the pure sugar, that is lactose, sir.
Okay.
Here.
Okay. Thank you.
Thank you.
No question from my side. Thank you.
Sharad Gangavanai. Are you there, sir? Sharad Gangavanai.
Hello, ma'am. Hello. आवाज येत आहे मॅम?
हाँ येतोय. You can start your presentation, please.
Yes, ma'am. Just wait.
Ma'am, please you stop your screen sharing.
Yes.
Nausheen ma'am.
Yes.
Sharad sir, you can start your screen sharing.
Yes, ma'am. Yeah.
अजून एक आहे जयपूर.
Is it visible, ma'am?
Yes.
Okay, I'm opening through my WhatsApp. There is a problem, ma'am.
Your voice is very unclear, sir.
There is some problem. Just give me a minute.
You just start your video and speak, please.
Yes, ma'am.
Without PPT, you can start your presentation. No problem, sir. On WhatsApp, your screen is sharing, sir.
Yeah, there is PPT, ma'am, in WhatsApp. That's why I'm checking where is actually is that.
Please, you can start your presentation orally, sir, if you want to give.
Just within a minute.
Yes,
I will start,
please.
ma'am.
We are waiting, sir.
Yeah, ma'am. Yes, ma'am. I got it.
Please, you will get only 2 minutes, sir. You have to wind up it in 2 minutes.
Yes, ma'am.
Your voice is very low, sir.
Is it visible, ma'am?
No. Your PPT is not opening, sir.
I'm trying from my side, but there is a problem. Can I?
Zoom will not allow to WhatsApp sharing.
Zoom is not allowing to WhatsApp sharing, sir. If you want
You
to
can go
read
for
your
next presenter.
presentation, please start orally, sir.
Okay, ma'am.
We are already getting late.
Yes, ma'am. Yes, there is now.
Next
now,
is
ma'am?
Amol Patil.
Now, ma'am, is there screen? Is there visible?
Yes, you can go.
Yes. Go on.
Okay, thank you.
Go ahead, sir.
Yes. So, good evening all. Myself, Mr. S.C. Gangavane, research scholar in biotechnology, SRT.
You can directly go to result and conclusion, sir.
Yeah. My topic
Sharad,
name
sir.
is, ma'am, in vitro.
Your voice is very low.
Yeah, ma'am. My topic name is in vitro induction and analysis of fast-growing normal root culture in Flamingia tuberosa, a rare medicinal plant. Next, in this, we know that in India there are almost 17,000 species of flowering plants, and 18,000 species are claimed.
You can directly go to result discussion conclusion, sir, please.
Yes, ma'am. In Flamingia tuberosa, it has some.
Please directly go to result and discussion, sir.
Yes, ma'am. Yes. Just
Again and
please.
again, I'm requesting you.
Its roots and tubers are sweet in taste and edible astringents, and it can be used for because it has a medicinal property. So dysentery and diarrhea and vaginal discharges against it is used. And this species is actually endemic and endangered. That's why we have used for the further study. These are some photographs of showing the roots of Flamingia tuberosa and seeds and tubers and that objectives. So, okay, ma'am, I am come to the materials and methods. In that we have
conclusion.
Yeah, last, ma'am, means just okay. After the collection of that plant, and we have just okay. So observation and after the experiment, lastly, just I explain in that these are the some roots which are normal and fast growing.
Conclude.
The half ms medium and 1.5 milligram Na and 2 milligram of IBA, that is hormonal concentrations. In liquid media is also their roots, and these are some chemicals which are found in vitro raised roots and natural roots of the F. tuberosa. And these are the HRLCMS chromatogram for Flamingia tuberosa. And these are the chemicals we have analyzed by using this qualitatively and quantitatively. And near about, sir, 57 chemicals we have got by doing the analysis in this plant, which has a medicinal property. And summary and conclusion. So at the end,
conclusion.
Yeah, ma'am. Yeah.
Again and again, we are requesting you, sir.
Yeah, yeah. Within 1 minute, I will complete it. So, in population of F. tuberosa is rapidly decreasing due to its edible property, different human activities, and overgrazing. So, summary and conclusion, we can say that the half MS medium, along with the vitro seed germination, and also we can say that when you use the IBA and Na concentration in particular amount, it shows the maximum significant root induction, which is fast growing roots, and the highest of 70% in vitro seed germination was found in the half MS. Then
next is Patil Amol.
Okay, madam.
Patil Amol.
Yes, madam. Yes, madam.
Yes.
Madam.
2 minutes, sir. Yes, you are audible, sir.
I will finish my topic and conclusion within 2 minutes.
Yes, sir. Last presentation.
Please remove the screen share. Madam, पहिले ज्यांचा आहे त्यांचं screen remove करायला सांग. Okay.
Yes, you can start your screen sharing, sir. Amol Patil.
Yes, madam.
Go to conclusion, sir, please.
Yes, 1 minute, madam. 1 minute.
Yes.
There is problem in screen sharing. I will explain from my side.
Yes. Okay, sir. Thank you. You can.
Okay. 1 second, madam.
You can start your video, sir.
1 second. Myself, Patil Amol. I'm a research scholar in the Suresh Gyan University, Jaipur, Rajasthan. My title topic is Formulation and Evaluation of Lipid Nanoparticles for Solubility Enhancement and Improvement of Dissolution Profile by Spray Drying Technology. Basic objective of the study is that is something drug soluble is less soluble drug, that is Lornoxicam. We have to enhance the solubility. Different method techniques has to be adapted, that is spray drying technique. The Lornoxicam has been obtained from Aristo Pharma, Mumbai. I have selected that is basically 6 batches of formulation, that is molecule is Lornoxicam, and the excipient is Gelusavan cross-povidone. Method is adapted, that is spray drying methodology. Something parameters has to be said, that is quality by design has to be selected, that is 2 by 2. Totally basically 4 batches has to be done, and these solid lipid nanoparticles has to be implemented for the next, that is floating tablets. These SLN is converted into floating tablets. That is my part of conclusion, is that from the result of above mentioned experimental study, we have concluded that the present study demonstrates the high potential of spray drying technique for successful preparation of Lornoxicam Larnoxicam and lipid nanoparticles using Gelusar 44/14 and crosspovidone. It is also found that Gelusar 44/14 has given as a rapid release than that of crosspovidone, since Gelusar 44/14 has found as a suitable polymer for fast dissolve of Larnoxicam. And I'm using that is, I have prepared the SLNs, that is solid lipid nanoparticles, and these solid lipid nanoparticles has to be converted into tablet, and that is total 9 batches has to be done for the tablet, and optimize the batch has to be compared with the marketed formulations. Okay, thank you.
Thank you, sir. Thank you. Avinash sir and Mukund sir, you can ask questions, sir, if any.
No question, please.
Okay.
Patil Amol, your topic is like formulation, evaluation of lipid nanoparticles for solubility enhancement, improvement, dissolution. It's a very long topic. And
yes, sir.
What nanoparticles you used for the evaluation? What commercial, some you said commercial nanoparticles?
No, sir. Actually, this SLN is converted into tablet.
Are you present? I was very confused. It is written formulation, evaluation, solubility enhancement, improvement, dissolution, spray technique.
I have prepared SLNs for the improvement of solubility by method of, that is spray drying technology. Spray drying technology is basically used for the enhancement of solubility.
Hey, what do you want to explain from your presentation? What is the exact conclusion from your presentation? I'm a bit confused with your title.
Conclusion.
One line conclusion. One line conclusion from your presentation.
Okay.
Okay.
It's okay. It's okay.
On behalf of Organizing Committee of ABM 2022, I really thankful for Dr. Avinash Saudekar sir and Dr. Mukund Adsul sir for your time and support. Thank you very much, sir.
Thank you, Organizing Committee, for inviting us. Thank you, Dr. Avinash.
Thank you. Thank you for the inviting, ma'am. Thank you, sir. Thank you very much.
It was a nice presentation.
Thank you. Thank you very much.
Some presentations are very nice presentations.
Thank you, sir.
There are clear presentations, some of the presentations. So like the session. Thank you.